during this session. So there are already about 200 attendees who have joined in. So please go ahead with the session.
Okay, so I'm starting with the session.
Yes, ma'am.
Kalal sir, please take a video. Kalal sir, video is not on. Kalal sir, please share your video. Kalal sir, yes. Okay, sir. Hope it's okay now. Yeah. Hope it's okay. Okay, madam, you can start now. Ati madam, please.
Okay, thank you. Yes, sir. Thank you, sir. Excellency, General Secretary Dr. Ram Kulkarni, Principal Dr. Dhanesh Kala, distinguished guests, eminent speakers, my colleagues, and all participants. Good morning and welcome. I'm Dr. Meenakshi Rathi and I will be the moderator of today's webinar. Thank you everyone for taking time out and being here online once again to attend the national webinar on features of ChemDraw and greener aspects of drug discovery organized by the Department of Chemistry and IQAC of Berkeley Education Society, RNC Arts, JDP Commerce, NSC Science College, Nasik Road, Nasik, in collaboration with Association of Chemistry Teachers, ACT, care of Homi Bhabha Center, Science Education Mumbai, and Cube Scientific Software Solution, New Delhi. As you might know, we faced certain technical difficulties last time, which is why we had to postpone this program to today. I would like to take this opportunity to thank everyone for bearing with us and understanding the unforeseen challenge we saw in the previously scheduled program. I know everyone was very eager to listen to and attend this event. So without any further ado, I would like to invite the person behind this entire webinar, Dr. Sudesh Ghodera, to share a brief summary of this program. Dr. Sudesh Ghodera.
Okay, thank you, madam. I am Dr. Sudesh Ghodera, convener of this webinar on features of ChemDraw and greener aspects of drug discovery. For this inauguration function, I welcome Dr. Ram Kulkarni, patron of this webinar and the General Secretary of Berkeley Education Society, the Principal and Principal Dr. Dhanesh Kala sir, the President of today's inaugural function, Manjusha Kulkarni Madam, Vice Principal, Dr. Sanjeevan Kharath, Head of the Department of Chemistry Department of our college, the speakers, Professor Nathu sir, Professor Brijesh Paren, technical consultants from the School of India, and the Professor Dr. D.V. Prabhu from the Association of Chemistry Teachers. I am going to say about why we are organized this particular seminar in this particular time. So the theme of the seminar is, we are aware of that during the period of lockdown due to COVID-19 pandemic, the academic work is done from home and many topics of interest are discussed and addressed nowadays through the webinar. So we, the Chemistry Department of Berkeley Education Societies, RNC Arts, JDB Commerce and NSC Science College, Nasik Road, Nasik, Maharashtra, in collaboration with the Association of Chemistry Teachers and Perkin Elmer through their reseller, Scoop Scientific Software Solutions Private Limited, New Delhi, have planned to organize this national level webinar on the features of ChemDraw and greener aspects of drug discovery. The greener aspects of drug discovery will provide an opportunity to discuss broader training needs for the future. This topic will increase capacity of researchers for drug discovery, and their efforts may be continued to build a pool of expertise in the greener aspects of drug discovery area. It is also the need of chemistry researcher to use and draw molecular structures. ChemDraw is one of the software which facilitates the need of researcher in various ways. With its origin as a chemical drawing tool, it has evolved steadily to become the leading chemically intelligent solution for multiple disciplines from specially chemistry to pharmaceutical drug discovery. So it is again a need of making the researchers aware about the use of this software. This webinar is seeking to create awareness and address the features of ChemDraw software and to get familiarized with greener aspects of drug discovery, along with some highlights will be given by Professor Pare on the chemistry education. The research institutions, undergraduate students and postgraduate students and other beneficiaries of this webinar will take up the ideas discussed here for their research and engage themselves in the process of transferring knowledge and technologies. I am very happy to share here that there are about 1,342 registrations for this webinar. So I can say it has a successful webinar. Thank you, and over to you, Dr. Meenakshi.
Thank you so much, sir. 10 months into the discovery of COVID-19 virus, almost the entire humanity is under lockdown. This global pandemic has brought life to a standstill. It only briefly, thanks to our patron, Dr. M.S. Gosavi, Secretary and Director General, Dr. Mrs. Dipti Deshpande, Director HR, Shri S.M. Gosavi, Director Education and Management, and Zonal Secretary, Nasik Division, Dr. R.M. Kulkarni, who have always blessed us with an object of encouraging spirit that learning should never be stopped. So the Department of Chemistry and IQAC feel proud to host this webinar for me. I request our Vice Principal and IQAC Coordinator, Dr. Manjusha Kulkarni, to welcome our guest of honor, speakers, and also introduce our patron, Zonal Secretary, Dr. Ram Kulkarni. Ma'am?
Yeah. Good morning to everybody. I'm Dr. Manjusha Kulkarni, Vice Principal and IQAC Coordinator of our college. On behalf of Honorable President of this webinar and Principal Dr. Dhanesh Kalal, I welcome our guest of honor, Dr. D.V. Prabhu, General Secretary, Association of Chemistry Teachers. I also welcome our honorable eminent invitee speakers, Professor A.A. Natu, visiting faculty, ISER Pune, Professor Brijesh Pare, President of ACP, also Head Department of Chemistry, Government Madhav Science, PG College, Ujjain, Mr. Charitra Gaur and Dr. Saurabh Sinha, representatives of the Scoop Scientific Software Solutions, Delhi, invited guests, teachers, and student participants from all over India in this national webinar. I take immense pleasure in introducing to you our Zonal Secretary of Gokhale Education Society, Nashik Zone, Honorable Dr. Ram Kulkarni. He is presently Principal of Gokhale Education Society's DYK College of Commerce, Nashik. Principal Dr. Ram Kulkarni is a renowned educator who has been awarded with over 25 awards. To name a few: Ideal Teacher, Nasik Road Bhushan, Ideal Social Worker, Bhima Ratna, and many more. He has authored about 71 books and has been an editor for 30 books. He has successfully organized and worked as director for more than 75 conferences, including 3 international ones. He is also very active and dedicated to his research work. About 11 students have been awarded PhD degrees under his guideship, in addition to the 3 MPhil students. He has presented number of research papers in international conferences in Thailand, Malaysia, Singapore, Dubai, etc. We are blessed under his enriched, able guidance. Thank you. Thank you. I am now
Thank you
request
so much,
Rathima.
ma'am. Thank you so much, ma'am. Now
Thank
let us have the pleasure of listening to the opening remark and guidance by Dr. Ram Kulkarni. Sir. Sir, Ram Kulkarni.
Video.
Dr. Ram Kulkarni, sir, please speak.
Sir, you are not audible.
Ram Kulkarni, you're on mute. Please, can you unmute yourself?
हॅलो, तुम्ही म्यूट आहात सर. तुम्ही आवाज चालू करा. म्यूट आहात. तुम्ही अनम्यूट करा.
There is some technical issue.
Ram Kulkarni, you can speak now, sir. You're unmuted. Can you speak now, please?
इथलं बटन सांगू का सरांना तिथे असलेलं?
Kulkarni, sir, please speak.
तू सांगते का बोलते का फोनवर सांगते का ते
We
बोलते?
can see Dr. Ram Kulkarni, but I guess there is some issue with is not audible.
हॅलो, हा सर. व्हिडिओ केला त्यांचा.
No, Dr. Ram Kulkarni is offline now. I can see he's offline.
Can we move on? Can we move on to the next presenter? So, can we choose the next panelist before?
Yes, when he joins back, then he can speak. Otherwise, yeah.
Perfect. Yes.
Okay, so shall we start with the next?
Yes, ma'am. Please.
Yes.
We feel privileged to have distinguished guest of honor, Dr. D.B. Prabhu, with us. And I request Dr. S.B. Ghoderao to give brief introduction about his accomplishment and attainment. Dr. Ghoderao, sir.
Yes, madam. Once again, good morning to all of you. I am going to introduce the guest of honor of today's inauguration function, Dr. D.V. Prabhu, sir. Dr. D.V. Prabhu is an adjunct professor in the Wilson College, Mumbai. He has published more than 51 papers in the various journals and 80 presentations in the conferences. His research areas of interest are reaction kinetics, catalysis, and environmental chemistry. He is the chief editor of Globalized Research Journal of Chemistry and Green Chemistry and Technology Letters. He is also the member of the editorial boards of Rasayan, Journal of Chemistry, and International Journal of Applied Chemical and Biological Sciences, Saudi Arabia, and Education in Chemical Science and Technology, the Journal of Indian Chemical Society, and he has authored in physical and analytical chemistry. He is actively involved in the training and mentoring of students for international chemistry and junior science volunteers. He is the chairman of Bombay Association for Science Education, and he is the founder general secretary of Association of Chemistry Teachers. Under his guidance, 20 years for the establishment of Association of Chemistry Teachers has completed, and so under his able guidance, the association and the ACT has grown to a very large extent. I welcome Dr. Devi Prabhu sir and say a few words. Dr.
It is
Prabhu
my
sir.
honor to now invite the Honorable Dr.
Good morning.
Prabhu
Am I
sir
audible?
to guide us.
Yes, sir. Yes, sir. You are audible.
Morning.
Prabhu sir, you are audible. Yeah. Please speak.
Professor Nato, Professor Brijesh Pare, Professor Sudesh Goderao, all my dear friends. I cannot see you, but I hope you can hear me.
Prabhu.
At the onset, let me congratulate Professor Sudesh Goderao for planning and organizing this national webinar on a very important topic, features of ChemDraw and green aspects of drug discovery. The topic is useful to students, teachers, and researchers who use ChemDraw on a regular basis while writing their research papers and thesis. Drawing of chemical structures is always a difficult task that requires software like ChemDraw. In this context, the present webinar becomes very, very important. In recent years, green chemistry, based on the 12 principles given by Paul Anastas and John Warner in 1990, is being increasingly practiced all over the world. Training of chemistry is done in all labs with the help of green reagents, green solvents, and green catalysts, which increase the atom economy and thus prevent waste and pollution. One of the important principles of green chemistry is to use catalysts in preference to stoichiometric reagents, which are always used in excess. Green catalysts like enzymes, nanocatalysts, nanocomposites, and phase transfer catalysts are eco-friendly as they minimize the waste products and therefore thereby improve atom economy and the yield of reaction. Drugs are now being manufactured using green routes so that there is no harm to the environment and also the yield is optimized. I am glad that in today's webinar, we have very eminent experts speaking on their areas of expertise. My good friend Professor Brijesh Pare, President of the Association of Chemistry Teachers, Professor A. Nathu from ISER Pune, Dr. Saurav Sinha, and Dr. Gaud. I would like to take this chance to say a few things about Association of Chemistry Teachers. It is the apex national registered body of chemistry educators of India and regularly organizes conferences, workshops, and symposia for teachers, researchers, and industry. association organizes an annual National Convention of Chemistry Teachers, and so far we have organized 19 such conventions. The association has also organized 3 international conferences in collaboration with Royal Society of Chemistry, London, UK. We have also organized about 20 chemistry popularization workshops with the help of DST, and Professor Pare was the convener for this program. We have also organized training workshops for teachers under the Dr. Yusuf Hamid Chemistry Popularization Program in collaboration with Royal Society of Chemistry, UK. For the students, we celebrate December 10th as a National Chemistry Day by organizing chemistry conferences, competitions for school and college students. One of the important activities is the concept test in chemistry for our undergraduate students. It is possibly the only test for BSc students in our country. We also publish a quarterly newsletter, which is there on our website. And every year, we give 8 awards to recognize the outstanding teachers of our country. This year, we are starting a new award, that is the ACT Award for Outstanding Contribution to Research in Chemistry. These awards are given after a screening, and so far, we have been able to honor more than 55 teachers all over our country. It is a matter of pride for us that all the leading chemical scientists are honorary members of our association, including Bharat Ratna Professor Siana Rao, who is our patron and the guiding spirit behind all our activities. The Association of Chemistry Teachers also gives academic support to a peer-reviewed chemistry journal, Globalized Research Journal of Chemistry, which is abstracted and indexed in Chemical Abstract CAS USA. And we also have academic support to another journal, Green Chemistry and Technology Letters, which is published from Muscat, Oman. So these are some of the activities which we are doing. I urge all the teachers to become members of our association and participate actively in chemistry activities. The application form is there on our website. Please refer, fill it in, and send your application form. With these few words, let me once again congratulate the organizers for organizing this timely seminar, and I wish the seminar grand success. Thank you very much.
Thank you so much, sir. Thank you, sir, for your words of wisdom. And it will be our great pleasure to listen to you again in our future.
Thank you very much, madam. Thank you.
Thank you, sir. Ram Kulkarni, sir, is he available here?
Madam, I cannot see Ram Kulkarni is online. He's offline. He's not there in the meeting now.
So, okay. So, shall we proceed?
Yes.
Or Dr. Manjusha Kulkarni, ma'am, you just gave him a call. Otherwise, I will continue. Or do one thing. I will first.
Dr. Meenakshi, we can go ahead, and then once Dr. Ram Kulkarni joins in, then we can have the session added.
Okay. Okay, no problem. So, Manjusha Kulkarni, ma'am, I'm taking the next part because she is going to introduce the next speaker.
नाही सर, तुम्ही लगेच जॉईन व्हा, मी सांगते तसं. सांगते तसं. ओके. हॅलो. हा,
Is he coming?
हा मॅडम, नाउ ही इज जॉईनिंग अगेन. ही इज जॉईनिंग अगेन.
Okay.
आर्या.
Our Zonal Secretary, Dr. Ram Kulkarni sir, is joining us. Or ma'am, I guess we'll take Principal sir's
Speech.
introduction. Yes.
Yeah.
Okay. Not a single event can be performed without a leader and his continuous efforts. He is none other than our principal, Dr. Dhanesh Kalal. I request Dr. Manjusha Kulkarni to give brief introduction of sir. Ma'am.
Yeah. Thank you, Rathe madam. Now I would like to introduce you Honorable President of this webinar and Principal of our college, respected Dr. Dhanesh Kalal. In addition to PhD in Geography, he is also qualified as a Master of Business Administration, MBA, MMS and postdoc. He is a very good administrator and geographer. He is also recipient of DAD Fellowship for postdoctoral research. He is serving since past more than 35 years in our prestigious Gokhale Education Society. Now, I am happy to say our society has just completed 102 years. He is also the branch secretary of Gokhale Education Society's Jawhar Zone. He has also worked as peer team member and NAC coordinator of NAC Bangalore. He has many research papers published in national and international journals to his credit. We are fortunate to have him as a leader. With this few words, I take your leave. Thank you very much. Now I hand over to Dr. Meenakshi.
Now I would like to invite Principal Dr. Dhanesh Kalal sir to address the gathering in his residential note. Sir. Sir, you are not audible. Kalal sir. Kalal sir, you are not audible.
Yes.
Dr. Ram Kulkarni has joined meeting.
Oh,
Okay.
So
I guess
meanwhile,
there is some issue with.
Yes.
Rathe madam, can you ask Ram Kulkarni sir to speak, please?
Yes. The guest of
I guess
honor.
let me.
The guest of honor of today's webinar, Dr. D.S.
Yes.
Prabhu, the General Secretary of Association of Chemistry Teachers, the Principal of Nasik Road College, and my colleague Dr. Dhanesh Kalal, Vice Principal Dr. Manjusha Kulkarni, Head of the Department Dr. Sanjeevan Kharath, the convener of this national webinar, Dr. Sudesh Ghoderao, Dr. Mrs. Meenakshi Rathi, Professor Nathu, Professor Pare, and all participant friends. I'm very happy to be amongst you this morning for the inauguration function of the national webinar organized by the chemistry department and the IQAC of Nasik Road College. Now we are in the midst of the pandemic situation, and so webinar is a perfect alternative to the seminar, because nowadays we cannot organize seminars. The Nasik Road College has a big tradition of organizing seminars on different subjects, especially in Golden Jubilee era of Nasik Road College. There was a tradition of arrange one seminar every month, and approximately 60 seminars were arranged during the period of 5 years. And I'm also very happy that Dr. Manjusha Kulkarni, Dr. Ghoderao has organized this particular national webinar on the theme of features of ChemDraw, the green aspects of drug discovery, which is a very relevant topic for all the chemistry academicians and chemistry students. I appreciate the efforts taken by Dr. Dhanesh Kalal, Dr. Manjusha Kulkarni, Dr. Kharat, and Dr. Sudesh Ghoderao for organizing this national webinar, which is viewed by more than 1,000 participants. I also congratulate Dr. Dhanesh Kalal for organizing this national webinar on behalf of the Nasik Road College, and I extend my best wishes for the valuable discussion in this seminar. Thank you very much.
Thank you, sir, for your blessings and best wishes. Thank you so much. Now I guess Dr. Dhanesh Kalal, sir. Now I would like to invite Principal Dr. Dhanesh Kalal to address the gathering. We are not able to hear you, sir. Kalal, sir, you are not audible.
Kalal sir, please speak.
It's not audible.
Kalal sir is not there in the meeting.
Okay.
Can I read his message in that case? I think we should read his message. His speech is there.
At least,
yeah.
Yes, sir.
So I'll give the message from the principal. Good morning to all of you. It's happening to know from Dr. Sudesh Ghoderao, the convener of this webinar on features of ChemDraw and greener aspects of drug discovery, that more than 1,300 students and teachers have registered. I would also like to put on the record the theme, which in fact is the need of an hour, as rightly said by the Dr. Ram Kulkarni, General Secretary of the Gokhale Education Society, in his introductory remarks. Mr. Charitra Gaur and Dr. Gaurav Sinha, technical consultants of Scoop Scientific Software Solutions, will give very valuable inputs in the field of ChemDraw, a software tool used in drawing structures of chemical components. Also, the resource persons like Professor A. Nathu of Indian Institute of Science Education and Research, Pune, and Professor Brijesh Pare, President of Association of Chemistry Teachers and Head of the Department of Government Madhav Science PG College, Ujjain, who will give altogether a new approach to know and implement new approach of drug discovery, which will guide us to know environment friendly production of drugs, resulting in lesser production of effluent, which will contaminate air and water. Lastly, I would like to thank Professor Sanjeevan Kharath, Head of the Department of Chemistry, Dr. Manjusha Kulkarni, Vice Principal of Science Stream of this college, and all the efforts taken by Dr. Sudesh Ghoderao to make this webinar a success. I am sure that each and everyone present for this webinar would have been immensely benefited by the valuable inputs, which will surely help them to develop interest in the areas of the greener aspects of drug discovery. Thank you. Kalal sir has joined the meeting again.
Hello. Hello.
Please, sir, speak.
Yes, sir, you are audible.
Yes. I'll just add few things to what Professor Ghoderao has read about my presidential address. Dr. A. Nathu, Professor of IISER Pune, Dr. Brijesh Pare, Professor, Association of Chemistry, Dr. Anindya Ghoshal, Organizer, Dr. Meenakshi Rathi, Panelist and Computer, Principal Dr. Ram Kulkarni, Zonal Secretary, Oakley Education Society, and former Principal of this very college, Dr. Saurabh Sinha, Dr. Subhash Sukumar, then our convener of this webinar, Dr. Sudesh Ghoderao, Vice Principal of this college, Dr. Manjusha Kulkarni. Our guest speaker, Dr. Prabhu, gave a good gist of this webinar, and it gives me great pleasure to put on record the efforts taken by the head of the Department of Chemistry of our college, Professor Sanjeevan Kharath, and the responsibility undertaken by Professor Sudesh Ghoderao in the conduct of this webinar named Features of ChemDraw and Greener Aspects of Drug Discovery. Our college functions under the management of Oakley Education Society, of which Sir Dr. M.S. Gosavi is the Secretary and Director General, and Principal Dr. Mrs. D.P. Deshpande is the Honorable Director, Human Resource of the Oakley Education Society. I am proud to say that our society has completed 102 years of its meaningful existence, rendering service to the masses, educating people, giving value-embedded quality education at an affordable cost. In the capacity of the principal of this college, I heartily thank Dr. Mrs. D.P. Deshpande, Honorable Director, Human Resource, for generously giving permission to conduct this webinar. Principal Dr. Ram Kulkarni, Zonal Secretary, Branch Secretary, and former principal of this college, has, in his introductory speech, has very nicely explained the importance of this webinar. It's heartening to know from Dr. Sudesh Ghoderao sir, the convener of this webinar, that more than 1,300 students have participated. I would also like to put on record the theme of this webinar, which is in fact the need of the hour. And Mr. Charita Gaur, Dr. Gaurav Sinha, technical consultants of Scoop Scientific Software Solutions, are going to give very valuable inputs in the field of ChemDraw software tools used in drawing structures of chemical substances. I also understand that resource persons like Professor A. Nathu of Indian Institute of Science Education and Research, Pune, and Dr. Brijesh Pare, President of Association of Chemistry Teachers, shall give us a new approach to know and implement new ways of drug discovery and guide us to know the eco-friendly aspects of the production of drugs, resulting in low generation of effluents. I am grateful to all other speakers who are present today, and I'm sure this webinar will benefit to all participants in a meaningful manner. Thank you. Thank you.
Thank you, sir, for your wishes and very influential message. Thank you so much, sir. Now
I would request.
here inaugural we have.
I finished.
Yeah. Thank you, sir. Thank you. Now we have just finished the inaugural function. Now it's the time for the gratitudes. Gratitude is the attitude that takes you to your altitude. So it's my privilege to propose vote of thanks for this inaugural function. At the outset, I would like to express my profound gratitude to Dr. Ram Kulkarni, Zonal Secretary, for sparing his time to address the gathering. We have been fortunate enough to be backed by very motivating principal, Dr. Dhanesh Kalal sir. I must mention our deep sense of appreciation for sir for always motivating and guiding us. I take this opportunity to extend our most sincere thanks to our guest of honor, Dr. D.V. Prabhu, for gracing the function and sharing his views on green chemistry and also explaining the contribution of ACT for honoring the chemistry teachers. I also wish to express my gratitude to Dr. Manjusha Kulkarni for always being with us and for her guidance. Thank you, Dr. Sudesh Ghoderao, for organizing such a thoughtful and thought-provoking webinar. Thank you all the eminent speakers and guests who are with us today to guide us. We are also grateful to all who have accepted our invitation to be the part of this event. Thank you so much. And immediately we are starting with the technical sessions. Before starting the technical session, I just want to tell you about today's webinar, how we are going to proceed. We are starting with the technical session 1 immediately. Then we will have technical session 2 on the topic, the significance of chemical revolution initiated by Loisier. And then technical session 3, that is on features of ChemDraw and software. Now, welcome back. Now we are entering in the first technical session of today. I request Dr. S.P. Ghoderao to introduce our first eminent speaker, Professor A.A. Nathu.
Thank you, Reddy Madam. Now, I'm going to introduce the speaker of the first technical session, Professor Arvinda Nathu. Professor Arvinda Nathu is a visiting faculty at IISER Pune. He is a senior scientist for last 40 years in National Chemical Laboratory and IISER Pune in the organic and bioorganic chemistry. He has taught at postgraduate level in the IISER as well as at the various institutes in India and in Germany too. The courses he has taught are biopolymers, separation of techniques, organic chemistry, bioorganic chemistry, and drug discovery. His research interests are organic chemistry, bioorganic chemistry, combinatorial chemistry, and biology in the drug discovery. He is honored with the various awards And to highlight few of them, he has awarded with the Cross Order of Merit, that is the highest civilian award for foreigners by the German president. He is involved in planning and execution of ICER Pune project and ICER Tirupati of MHRD Government of India as a task force member. He served in pharmaceutical industries in India and abroad in various capacities as director, consultant in discovery, process development and management, and IPR, etc. He is involved in Indo-German collaborative activities in science, education, and technology. He is the leader in Indian Chemistry Olympiad teams in Budapest, Cambridge, and Georgia, and came out with the flying colors. Some of the awards and honors to his credit are: he is a Honorable Advisor and German Research Ambassador since 2007. His lifetime award achieved award by Labdi Research for Development and Education RD of rupees 1 lakh awarded by Labdi Foundation, Gujarat, India. He has a Lifetime Achievement Award awarded by a consortium of North Maharashtra University titled Golden Opportunities for Latest Developments in Chemistry and Technology. He has Lifetime Achievements Awards Suryadatta Educational Institutes, Vijay Joshi Trust, rupees 1 lakh, and Buffett Millennium Institutes. He's been associated with the he's got the Vocational Excellence Award by 2 Rotary Clubs. He led the Indian Chemistry Olympiad teams of India in Budapest, Cambridge, and Georgia and came out with the flying colors. He has got the Best Teacher Award by PVG Group of Pune. He is a fellow of Maharashtra Academy of Sciences. He is a member of Board of Directors of Pharma companies and PCMC Science Center. He is a scientific advisor in the area of education and health of female in rural area and scientific research. He is a member of Academic Council of Pune University. He is the ex-member of Senate of SV National Institute of Technology, Surat. He is the ex-member of Academic Council of D.Y. Patil University, Pune. He is the founder member of Indian Peptide Society and Indian Society of Bioorganic Chemists. He is the member of National Steering Committee for Olympiads in Homi Bhabha Center for Science Education. He is a visiting scientist, Bielefeld University, and is the adjunct professor at SP Pune University. The other important activities in which he is involved is involved actively in the underbringing of the underprivileged tribal, female, and northeastern students to the mainstream of education and health through camps and lectures and hands-on training, etc. So with this brief introduction, I welcome Professor Nathu sir and to speak on his title. Thank you.
Thank you so much, sir. We rarely get this kind of opportunity to listen to the words of great personality like Dr. A.A. Nathu. So I invite Professor A.A. Nathu. Sir? Nathu, sir?
Now, where is the sharing window? I cannot find.
Where
Where it
desktop is visible. I think your desktop is visible.
You can see.
yeah, it is visible.
Yes, it is visible.
Where I can start it. This is the my presentation, but there is no. I don't see any share. This one.
Your screen is already shared. Please start your PPT. Just click the PPT that you want to share.
Okay. 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 Is it visible now?
Just click that PPT again completely, sir. Just click the PPT. You just double click it. Yeah, and maximize the screen. And
is it okay now?
Yes, sir. It is visible now.
Oh, should I start? Okay.
Absolutely, sir.
Yeah.
Go ahead.
Should I start then?
Yes, sir. Yes, sir. Go ahead.
Okay. Okay. First thing, thanks a lot for giving me this opportunity to opportunity to interact in this pandemic of COVID-19, where there is a lot of turmoil going on drugs and whatnot. I don't want to get into that mess, maybe at the end of my talk. Today I'll be talking about the green perspective in drug discovery. Mainly the drug discovery process is such a long process that it takes, it is now getting murkier and murkier into the chemistry business. By the way, I start with this slide always. This is where we started in 2006, the ICER Pune Institute, and you'll be surprised to know that we started with 10,000 square feet, maybe less than that. And today at the end, I'd like to show you how prolific campus of 4 lakh square feet we have. This is my mail ID. I'm approachable all the time through mail. Let's see how the chemistry is looked upon by the people. What is the perception of chemistry people have? It's very bad. That is because it's pungent, it's burning, hazardous, nauseating, and it is one of the biggest pollutant that you talk to world, you utter about the chemistry. The first thing that these 4 or 5 feelings will come to the mind, but chemistry is everywhere. Yeah, chemistry is everywhere. There is no place in this world where there are no chemicals. Of course, there is one which I would like to keep secret, and especially for those students. There is no other place where there are no chemicals. And secondly, if you go to the dictionary, meaning of chemistry is very, very variable. If you see here, the picture of the world meaning of chemistry, it is love, affection for each other. If the boy and girl meet, they never say our physics is matching or mathematics is matching. Everybody says our chemistry is matching, therefore. So can we convert all this business of perception of chemistry into this wonderful reality of chemistry? And that is the major issue, that this picture needs to be taken. I don't want to go into the greener details, green chemistry details, because so much has been said, several books have been published, but just to go through, first thing is to prevent the waste. Waste is such, at least in drug industry or pharma industry with which I am concerned, the amount of waste you have is about 2.5 kilos per kilo of the drugs. So can we afford to have that? Can we have atomic economy? Atomic economy, that means number of atoms placed in the starting material are fully converted like chemistry. Can we use ambient pressure and temperatures? Avoid the use of auxiliary substances such as solvents, separating agents, and all these things will add to the waste. Energy requirements, renewable raw material feedstock can we use? Reduce, can we use derivatization? Can we use catalytic reagent as selective as possible, better than the stoichiometry? Analytical methodology real time. Right now, there are some improvement on the analytical method, but what used to happen? You do the reaction, take out the analyte, and finally we'll come to know that all things have gone bad, so you need to waste it completely. But now there are many online methods with which you can test. Moment you think that there's something wrong, you stop it and go ahead. And can we have substances with minimized potential of chemical accidents, including releases, explosions, and gas? Okay. Now practice, there are several practices. I don't want to go into detail. Just after just saying that now when you say green chemistry, that we throw off phytochemicals photochemistry, sonochemistry, microwave. By the way, let me tell you one thing. People who are using these techniques are unable to explain why it happens. Similar thing, there are many catalysts used. Most important, can we use a solvent minimization? I'll go through some of the examples. Can we use supercritical carbon dioxide, which is the most friendly as far as the natural product is concerned? Can we use aqueous hydrogen peroxide, water reactions, dry media reactions, and so on and so forth? Can we avoid the use like HDM bromide, which is generally used in biochemistry so much, xylene, mercury, and so on. Now coming back to the drugs, I'll restrict myself to drug discovery is a big topic, and I think I run a 42 lectures course on this on drug discovery. But basically, to start with, there is a pool of compounds, and from these compounds, there is a preliminary biological testing that leads to lead generation, leading to toxicology, radio analysis, and all the things. Basically, this part takes more time in the whole drug discovery process. And then this leads to chemical manipulation and final compound. And this is a very, very lengthy process. What you need basically is more than 300,000 compounds. This is too less as compared to today's studies. Today, you need about 700,000 compounds to be prepared to come across one. Then you go in for biological activity, basic biological activity, before you make a drug application. And then comes the real problems and real trouble. Then it is totally in the hands of biologists or toxicologists. First thing is toxicology, how dangerous it is. That is number one. Your drug may be useful, but how dangerous it is. Second is bioavailability. How much is available at the site where it's designed? If you take a 500 milligram crocin tablet, paracetamol tablet for headache, probably what you need is not more than 5 milligrams. And why we take 500 milligrams? Because it spreads and then hardly 5 milligrams reaches to the site where it is required. So that is very important. Then there is a pharmacodynamics. What happens to this compound? That means if you take a compound, it doesn't stay in our body like this. It gets degraded, it gets changed. All kinds of reactions happen. What are those happen? And second, last is pharmacokinetics. How the drug is effective. So pharmacokinetics is something that the drug does to the body, and pharmacodynamics is what body does to them. So these are the two simple aspects. I don't know about it. Then it entered a human trial. This word is now really very common even in newspaper and WhatsApp about the human trials, which is a long process, and ultimately it is the FDA clearance, US FDA clearance, which is required. What is the time taken for all this business? Time taken for all this business is about for this about 14 years or 15 years. Believe me, the new drug that you get across the counter is discovered, the compound is discovered 14 years back. And this 3,000 is an old figure. Now it takes about 8,000 to 10,000 crores rupees for development of one. And then also you are not sure whether you are going to get a new drug or not. So this is in short. You need to synthesize array of compounds. You find out the suitable screening system. Most active is then only the thing is admit I'll go into these details. This is in short what I was talking to you. interaction of drug with the receptor is called the dynamics, and which metabolize is get into kinetics. What happens to a drug here is it get absorbed, it get diluted, it get diluted, and it get eliminated. These are hydrophobic part. Hydrophobic part is required so that the entry into the cell is easier, whereas the hydrophilic part is required because of the solubility of water. You don't worry about the big structure. Only remember a drug has to have balance between hydrophobic part for the cell entry and a hydrophilic part for their water solubility. And the whole fight or the balance is between the two factors to come. I don't want to go into these details right now. What I'm going to cover under drug discovery is only 4 points or 3 points and take the time. One, we talk of interdisciplinary things all the time and saying that the biology and chemistry have to go together, chemistry and maths have to go together, and so on. But in practice, we see we sow the seeds in the 11th standard when the student has been asked to draw biology. This is one thing that I would like to bring it to you. So I'll show you 2 examples, very simple examples where the biologist have helped the chemist to come across how without biology they would not have been working out in drug discovery. And next is how DNA and proteins are used as drugs, and this is where the chemists have come to them. So first is biologist help to the chemist, whereas the another is chemist help to the biologist, and this is all to avoid the chemical wastage and to bring as much as greener part as we go on seeing this thing. And then I may not touch chiral drugs. If the time allows, I'll do it. And I'll touch flow chemistry because now that is the hottest topic in drug discovery or drug preparation for that matter. So basically I plan to cover 1, 2, and 3. If the time allows, I may go in for chiral drugs or anything that I don't want to waste time on this. Biotransformation in industrial applications are very well established. Thousands of industrially important stains are available, are used for the production of several chemicals. They are non-hazardous, cost-effective, and so on and so forth, especially when they are working in aqueous media. But the real problem comes. You have a problem. For example, this is a statin intermediate I wanted to make of this sort. Even if you don't worry, if you don't remember the structure, what I want you to remember, there are 3 acetates around one group, and I want to hydrolyze only one acetate. It is impossible to use any chemical means to do that, and ultimately enzyme has proven to be very enzyme came to your rescue, and ultimately you got one hydrolyze. You protect it, then again use the enzyme. Use the enzyme and again get one of the 2 hydroacetoxy selected. And by converting this, you end up in this intermediate, which otherwise would have been impossible. Similar thing is one of the deltaism intermediate drug used for cardiovascular. Here, if you see here, there are 2 alcohols and 2 acetates which are converted. Using enzyme, you can isolate both the plus R isomer as well as the S isomer, which can both of them can be taken to deltaism. Here, I would like to show how the chemists have failed to do that. You want to convert, this is also a very, very important intermediate in the prostaglandin. This looks very simple. I want to convert this compound into this compound. Very simple. How it is done? You use caronaldehyde to resolve this, And this is again done by separation of a chromatography hydrolysis. Why? There is a high cost of chronaldehyde. If you want to make the chronaldehyde, you need to use another ozonolysis process. So make it in terms of making the greener aspect, you are inviting more and more trouble. How the biologists have helped? They have simply taken this diacetate, enzymatically desymmetrize it. That means once this acetate and once you get this acetate, and then using several steps, they are finally taking it to the yield or overall yield with a 50% and enzymatic or the diastereomic yield is very high. So this is how the enzymatic people, I just shown it just to show you, you get 74% yield and 98% enantiomer excess, and rest of the material can be recycled. This is really green chemistry. That means you are recovering the whole material, you are not wasting any time, and that too under completely aqueous medium or aqueous. Believe me, without biologists, this would have been really impossible. The real problem with the enzymes is enzymes are soluble in water, and the organic compounds are soluble always in organic solvent. So this was the thing. But now they have overcome this by saying what they do is they take the enzyme and dissolve it in buffer and completely remove the water. Then there is you may believe that there is a complete layer of a pH buffer. So enzyme remembers that buffer, and when it is reacted in organic solvent, this is done. So this molecular layer of water or the desired pH is a must, and now this is plentifully used in organic solvent. So problem of using enzymes in organic solvent has been now solved. Now let's go to the next one. I'll give you one example of how this is done. How a purely done of the green chemistry. Take this simple aldehyde, there's a drug called venlafaxine. Take this simple aldehyde and convert this into CH2CN, and then I wanted to convert this into this cyano derivative. What are the conditions? You have to use LDA, you have to use butyl lithium, you have to use anhydrous THF and minus 70 degrees. So jokingly, somebody told me, our gurus used to tell, why don't you go to Himalayas or Antarctica, do this reaction? At least you will save expenses on the cold. So this was the procedure. So now you see, and then treating with alumni, get this intermediate, and ultimately you get venlafaxine. So what is the problem? Problem is you use strong bases like butyl lithium, LDA, and how much solvent you are spending it. Secondly, low temperatures, minus 70, minus 80, anhydrous solvent, look at anhydrous THF, expensive reagent. So this becomes a completely different non-industrial process. How this has been overcome? You will not believe by this such strong bases, LDA, butyl lithium, and this reaction is simply done by the sodium hydroxide in water, and you will be able to get the desired product. Similarly, there is in one step this has been converted. So I can say this is really green chemistry, a dream run. The price, ultimately price of venlafaxine has been reduced, you believe me, to 1/10 to the manufacturer. I don't know how much it goes to the customer. The reaction can be performed by simple basis like sodium hydroxide. Process is very easy to perform on large scale. Avoid the use of hazardous and green solvent. This is a drastic cost reduction. This I consider to be really example of greener aspects in drug discovery or drug development. I don't want to go into details of this. This is good. Okay, now yes, now the biologist I have. Now let's see how the chemist I have biologist. This is a non-toxic ideal drug, does not interfere with the body metabolism, easy synthesis, easy scale up, commonly commercial made from body-like material. I'll come to that. Later. And avoid high waste, as I said, 1.5 metric this may in discovery and 2 kilos in clinical trial for 5 kilo of the drug. The real problem is ideal drug never exists. So I would like to concentrate more to look after. Can we? I'll first touch upon the DNA. There is a time cover on a special issue, say drugs of the future. Amazing new editions will be based on DNA. You'll be surprised how DNA can be used as drugs. Can we go back and see something? This is one drug, Kinamara, has been introduced in the market, which is completely a DNA-based drugs, and actually there are 6 drugs right now in the market. We need to understand a little bit how the DNA drugs work. This is a normal picture. I try to simplify it, considering that chemist has a back. And there is a bunch of chemists in the gather. You have a double-stranded DNA, you have get single mRNA, green one, and that leads to the production of protein. Production of protein, which is disease, which are basically diseased proteins. Which are diseased proteins. So if you have DNA belonging to the disease or the foreign DNA, right now COVID-19, let me put it this way, then what you can do is, before producing the these proteins have to do, and all treatments are given, or all treatments are given based on this disease protein, bad proteins, more proteins, less proteins, or whatever. But there are certain cases, especially the viruses, which has something called window period. What is window period? Please don't understand. You don't have to understand a lot of chemistry. Window period is that patient has the disease DNA, but is not getting expressed into the proteins. Now, how we can detect that? Can we do that? Okay. Bad at that. Okay. So I have a diseased DNA, and I don't, but proteins are not getting expressed in my body right now, and therefore none of the drugs or the diagnostic tools are working. So what to do? If you cannot catch them here, can you catch them at the intermediate stage of mRNA? Very simple trick. You know, every DNA, mRNA, everybody is complementary. So you make a small stretch of mRNA or DNA in the laboratory. mRNA or DNA in the laboratory, attach mRNA and DNA, go to the beans. Very simple method. You make and complementary the red shown DNA in the laboratory and send it in the body, and it will complement and things. Very simple way of doing it. But real problem comes, there are 2 issues. If you inject straightway DNA into our body, there are 2 issues. Number one is that because of the hydrophilic nature, as I told you earlier, this hydrophilic nature doesn't allow the DNA to go inside the cell. This is one of the major issues. And secondly, because it has to be hydrophobic. And secondly, most dangerous is once it enters, there are enzymes called DNA, they eat immediately hydrolyzed due to the cellular enzymes. So cellular enzymes hydrolyze, and the lipophilicity is not good enough to penetrate. So these are the problems. Biologically, concept was fantastic. But what to do because of these 2 problems? Then really here, the chemists came to the issue. What they did is very simple trick they made. Very, very simple trick. As you know, phosphorodiode backbone has all the 4 oxygen atoms. I have replaced one of them with the sulfur, called phosphorothioate. Once I make it phosphorothioate and allow it through the blood, then blood will not, or the DNA's enzymes will not identify this as a killer. And so this will go to the reach to this thing, and then some of them, you convert them into methyl phosphonate and impart hydrophobicity. So these are the two. What I did, I deceived the DNA's enzyme. I allowed the PAD entry. So very simple. These are the phosphorothioates. If you see two diastereoisomers, and I have methyl phosphonate. So I make a chain of DNA with some of them phosphorodiet, some of them with methyl phosphonate, as I shown you here, and make a compound, very simple compound. Now this DNA can go enter, because of the phosphorothioates, it won't get affected by the DNA's enzyme, and because of the methyl phosphonate. So this idea is now caught up, and a new drug, I can show you the number of modified structures here. Instead of phosphate, there is a phosphorothioate or methyl phosphonate, and you have number of drugs now coming in. One of them is Kinomara, is ready to fill in injection. Most of them are in the areas of cancer. There's another beautiful idea. This is a DNA where you had diphosphate as a ring. Can we convert this phosphate into the amide ring, same as peptide? And there are now many peptides that there are many compounds are available. They're called PNAs, that means protein nucleic acid, and they're also under trial where you can see. Now, once they got this idea that DNA needs to be modified chemically, then there was a plethora of compounds. I think there are more than now 5,000 such derivatives of DNA which are resistant to this bloodstream DNAs that are present. Therefore, this idea has worked out only because chemists have come to the help. Earlier, the biologists helped the chemists. This is how another chemist helped. Otherwise, a new completely branch of oligonucleotide would not have. I don't want to go into details of flow chemistry, these drugs, thyroid drugs. If I get time, I may enter. What I wanted to tell you is a new branch of medicine that is coming up, which is very easy. As I said, if your drug, if your body materials like drugs, DNAs, protein, carbohydrate, fats can be used drugs, they'll be always less toxic, and therefore an idea has regenerated, and this gives rise to an terminology of something called biological medicine. It is a medicine. Why I'm taking an English aspect, because this doesn't involve lot of waste. It is a medicine whose active substance is derived from living organisms. Insulin, IgG, protein-based drugs like somatostatin, and there are several drugs which are produced by living organisms. Then there are biopharmaceuticals. Drug is the same, but it is created by biotechnology means, especially by genetic engineering. You have genetically engineered several products such as insulin and so on and so forth. Now there is a biosimilar. It is a medicine which is similar to biological medicine that has already been authorized. But many times you are unable to get it, but you get a biosimilar or a similar compound to that which has got the same activity. And you may find it little bit offbeat to talk about it right now. But I can assure you, by year 2027-28, top 5 drugs or top 5 compounds or top 5 drugs of the 10 blockbusters will not be chemical, organic small molecules, but they will be either the biosimilars. DNA based or protein based. So this is how we would like to convert that into a completely green. And now this, I'm a little bit coming to a comparatively new technique that has been used now in organic synthesis in order to make much greener in many ways. Idea is very simple. You carry out your reactions always in a flask. You always carry the reactions in a. You carry always the reaction in a flask where A plus B gives you product plus X material. If you are atom efficient, then A plus B goes to the product. It can be a catalytic reaction, A plus goes to P, and or it could be A plus B goes to C and D, and so two reactions. Either it can be catalytic, either wrong. This we have been use of conventional wisdom tells us that can we have been doing this atomic economy and all other things. Only the thing is, can we really reduce the wastage using? Principle is very simple. You will feel that how you have not anchored it. Can we call or do all these reactions in a small capillary? In a small capillary, I'm not talking of the tubular reactor or those kind of things. I'm talking of a something called a catalytic reactor or a catalytic capillary tube. What I have is, I have capillary, this shown here, through which I inject a product compounds A and B with a small pump, of course, and then that leads to C. Then there is another small column through which the catalyst is packed, that goes to D, and ultimately this is separated and you get the product. Believe me, all these reactions are done in a small capillary. Earlier, they were as small as that. What is the sorry. Okay. So now how the reaction vessels look like? At the top, if you see this vehicle, this is can you see this small capillary is going on? This is one. Now, it's very, very simple. We create by water electrolysis, take the hydrogen, and instead of adding hydrogen, what I do is I keep constant and allow the solvent to flow around in many circles. So you can do this in small capillaries. I'll go into the details of this later on. You can do it this slightly bigger capillary, microfillet, this one, where the hydroxyl is converted into nitro, and so on and so forth. These are the series of pipelines now, or the series of tube reactors. Or the multi-reactor system, where now it is industrially also possible. And very simple, nothing more than that, nothing less than that. I don't want to go into it. What is the use? We'll see a few examples. What are the advantages of flow reactors? One is the thermal control. What reactions you can do at higher temperature, you can do it in lower temperature. Mixing efficiency is lower because the surface area is increased. Productivity and reproducibility is very high. There's a safety concerns are less in this, because for what you need about 1500 liters of vessel, you can do this with 150 liters of capillaries or tube. Or dangerous gases, liquid products, singlet, oxygen, you can do photochemical reaction, you do anything that you like. Let's go into few examples just to give you the flavor. The most notorious reaction that has been troubling us is the nitration of phenol, which always gives a mixture of these two, and yields are very high. It is a very exothermic reaction, so you have to do it at 55. Now, if you use this in tubular reaction, which I shown by the AMR this reaction, you can get the yields of 75%, 78% pure, 75%, 78% pure, and at 5 degrees centigrade. Can you imagine from 50 to 5 how it goes? There are people. I have ample of examples in low chemistry. I am just choosing very simple one. Small volumes you want. Let us take a very simple example of our alkylation using Ivan's chiral oxidation. If you do this reaction, very simple reaction, the alkylation here, you can see the regular batch process gives a yield of 31%, and the diastereochemic excess is always less than 85%, and then there are more than 10% byproduct. If you do the same reaction in flow chemistry, you will get 41% yield. Diastereochemic purity will be 99, and most important, the remaining material can be recovered. So this is really the green aspect of this flow chemistry. There are several reactions. You have a dibromo, convert this into monolithiated compound, substitute by electropile, and these by just using various pumps. You can see here using various pumps and various compounds, you can convert this into very, very high yields. You may feel this is a magic. This is not a magic. This is a regular chemistry and highly based on principle. You can do the photochemical reaction. Simply wrap the tubing around the light source through tube period. That means whatever your capillary is there, you introduce the lamp inside that. You can use the catalyst. Now you can use even the enzyme catalyst in doing this kind of thing. It can really do the multiple things. So this is a simple reactions of beta-lactam synthesis where you need reagent column, catalytic column, scavenger to remove the waste or whatever is unwanted, and the mixed column. Using various columns, I don't want to go into details of chemistry, it will take 1 hour. By using this mixture of various columns A and A, then you can go to put a catalytic reactor here and then scavenger column. Ultimately, you get very high yields of beta-lactam. This is a new synthesis has come in a year's time by a Japanese scientist, where artemisinin, a well-known artemisinin compound, is you need beta artemisinin methyl ether as the final compound starting from artemisinin, which is a natural product. And it was a very, very, very tough synthesis. And ultimately, by using pumps and the reactions, you can see here, O2, gas, liquid, everything is managed, and you get the beautiful yield of artemisinin. Let me tell you, let me remind you one thing. Just I cannot avoid of telling this fact. Ultimately, the artemisinin ether was discovered by a Chinese lady, Yu, and she discovered it in 1977, and now it has come to the market and so on. When I was surprised to know that she cannot speak English, so give a talk in Chinese language. Her paper was translated into English of late when she was declared the Nobel Prize. Just remind. So this is another compound, rolopram. You need to have chiral compound. So using again several pumps and several tubes, you can do this. Now, if you want, if this is a normal thing, And if you want to go into chemical reactors, you need to go such high. These are the chemical reactors that are now used for doing 50 to 100 grams of the product. Now, most of the high value products can do. It is used even in India in industry. So this is not a Jack and Jill story anymore. Why? Microreactors serve as a useful platform. Capillary reactors or microreactors. Number of differing microreactors have increased significantly in both form and composition. Ability to directly analyze the reaction provides quick optimization. Since this is a tubular reactor, you can connect your HPLC or GC to one of them and do a asymmetric catalysis is yet to be done. Reactions are limited to fast and issue with homogeneity. So, friends, what I have done, I have already taken more than half an hour. Perhaps I'll take another 5 minutes. Is it okay?
Yes, sir, it's okay.
I'll just take
It's okay, sir. Yes,
5
yes,
minutes more so that
yes,
let the others. Let there be any. So what I had taken to the wonderlands of three things: how the chemist have helped biologist, how the biologist have helped chemist to create the greener ways of doing the chemistry. Let's see, this is particularly these last two slides are meant for basically the student community. Students, what we have a rich biodiversity. We have a good synthetic organic chemist capable of generating large number of libraries of organic compounds. Excellent drug or medicine chemist, one of the largest pool of biologists in diverse branches. We are expertise in natural, we are very rich in natural product chemistry. We have availability of all supportive facilities now in most of them in bioinorganic or molecular modeling or X-ray crystallography and what. These are all branches that makes into it. And now in order to add, I may add the artificial intelligence and machine learning and drug discovery. That is also. But there's a big question. I am just putting few facts about the Indian pharma industry with which I am associated. And it's not a very happy affair over here. Present situation is about 2,300 drugs are made in India. 40% of their drugs are exported. You believe me, the drugs are the cheapest in this country. Your relatives, when they go to US, they must be taking a lot of paracetamol and ibuprofen and whatnot. That is because that is the cheapest in the world. But in the last 40 years, that not a single drug has been discovered for TB. Same situation with the parasitic diseases like malaria. Now, after 50 years, there is one drug, but that is not also in the main channel for TB and malaria to some extent. Yes. Why this has happened? This has happened mainly because these are poor man's or own ownership diseases. No foreigner will come and start a drug discovery on TB or malaria. That is because even if the drug comes out, who will pay for it? So, my dear students, it is our own heritage of TB, malaria. Believe me, today more and more, the most deaths in the country are because of TB, dysentery, and malaria. So we should have more disease, and who will do that? Who will discover that drug? It is you younger generations should be in it. Now the drug tendencies, earlier you remember we used to have penicillin, which is a broad spectrum antibiotic. Now narrow spectrum antibiotics are more familiar with you. For stomach, different. For lungs, you take lung infection, you take different. That is mainly again because the prices and the dollar runs that particular. What worries me more is what the red that I have shown here. Market is flooded with drugs for diabetes, obesity, various types of cancers, antidepressants, blood pressure, and psychosomatic disorder. Diabetes, we are the capital of the world, so I don't want to talk about it. Obesity, especially these antidepressants and psychosomatic disorder, is the donation of IT to us. We do not know how the IT has entered into the health and the social mentality of the people working there. 35 to 40% IT people as of today have either diabetic, blood pressure, or this. So this is a gift to it. What disgusts me more is these were all drugs were for export. By the way, psychosomatic disorders and antidepressant, these are the so-called mood elevators. Now at 12th standard, boys also asked, and boys said, I don't have mood. And then he's immediately getting taken to doctor. They are given mood elevators. And so what? I don't know what are all things by paying thousands of rupees. If I would have done this, my father would have just slapped the one I said, go to study. And that would have perhaps cured my all psychosomatic disorder. Forget about it. I don't want to criticize anybody, but this is a ground. What is more dangerous, we are entering into the mall culture. What is mall culture? You go to the mall, you want to buy a simple ball pen. And then what happens? If you want to buy a simple ball pen, you see that something is free on that, 2 on 3 free, 3 on that, free, cash back, and what all things. What happens? You buy unnecessary things what he wants to say. You don't buy what you want. Same thing is coming to a chemist. You go to the chemist and ask him for a simple that you have some acidity and ask him for some simple aluminum hydroxide. What he will do is, he will say, sir, you take this, but I'll remove your cause of acidity. I'll give you proton pump inhibitor. That sounds smart. He said, okay, give me. He'll give and immediately says, sir, you must be having nauseating this thing also. So you take better pantoprazole with domperidone. So you went there just to check your acidity for 50 paisa tablet, end up in paying 8 rupees per tablet. This is the mall culture where you don't buy what you want, but what is made available and what is sold. At least the educated people should be completely aware of it. And I'm thinking the whole audience is really a very good audience and they will do it. By the way, there is not a single drug has been discovered in India, and nowhere outside Indian or herbal drugs are accepted. So these are the two real, what you say, bad things that rumors in my mind, that with such a brand battery, we have not produced a single new drug. Neither our ancient way of traditional medicine has taken outside. You may be hearing stories about it, but believe me, nowhere in the country, nowhere in the world, our herbal medicines are recognized as medicines. So from 10,000 square feet, this is a 4 lakh square feet area we have come across. These are our classrooms where all modern things are there with air condition and lighters and top conditions and everything, except an old method of chalk and blackboard still they exist. Is the exist one. And our vision is very, very simple. Highest scientific caliber in terms of student training and scientific research. And I would like to profusely thank you for bearing with me my lecture for last 30 or 45 minutes. I didn't remember even what was the time assigned. I forgot to ask the organizer. I'm very sorry if I crossed the limit. Thank you very much. This is the end.
Thank you so much, sir. Attendees and participants, wasn't it really a very suitable topic in this current outbreak of COVID-19? Sir, you have very well explained the perception of chemistry, atom economy, and the steps involved in drug discovery, that is toxicology, bioavailability, pharmacodynamics, and pharmacokinetics. Even you have explained the human trials are not that easy, which are going on in this current situation. Thank you, sir, for explaining greener aspects of drug discovery, then characteristics of ideal drugs, as well as some drugs which make the future, drug of the future, and the biological medicine. Thank you, Professor A. Nathu. Now, we are entering into the technical session 2. The next distinguished to honor us with his presence is Professor Brijesh Pare. I request Dr. S.P. Ghoderao, please introduce our next eminent speaker, Professor Ghoderao.
Thank you, Dr. Rathi Madam.
Thank you, sir.
I'm very glad to introduce Dr. Brijesh Pare, who is actually my very best friend also. Professor Brijesh Pare right now is the president of the Association of Chemistry Teachers. Yes, having a teaching experience of more than 33 years at the postgraduate level. He is serving as a professor and head PG Department of Chemistry, Madhav Science Postgraduate College, Ujjain. He has received the fellowships for postdoctoral research from the National Research Foundation, Pretoria, from May 1999 to May 2000, to work in the Department of Chemistry, University of Durban, Westville, Durban, South Africa. He has to his credit more than 70 research papers in various referred international and national journals of repute. He has guided 18 PhD students and 6 students are registered and working actively for PhD right now. He is a member of IUPAC, that is the INSAV nominated affiliate membership IUPAC UNESCO for the term of 2012 to 2013. He is the coordinator of and organized more than 100 chemistry population workshops for the students and teachers across the India, Abu Dhabi, Dubai, Sharjah, Doha, that is UAE, and Qatar too. He is working actively with the Royal Australian Chemical Institute Australia and Royal Society of Chemistry London. He is the panelist as a panel member for national level Best Chemistry Teacher Award instituted by Association of Chemistry Teachers. He is the honorary coordinator of the and country head for ANCQ, that is the International Australian Chemistry Quiz, conducted annually by the Royal Australian Chemical Institute Australia. His research interests are science education, green technologies, and advanced oxidation processes. With this brief, and he has to his credit few awards, the postdoctoral fellow under National Research Foundation Pretoria and the INSA nominated affiliate membership of IUPAC. With this brief introduction, I request Professor Pare to speak on his favorable topic of the chemistry education. The name of the topic is the significance of chemical revolution initiated by Lavoisier. Thank you.
You are not audible, sir.
Professor Pare, please unmute
Not
yourself.
audible.
Professor Pare, you're not unmuted. Please unmute yourself.
Hello. Hello.
Yes, Professor
I'm audible now. I think it is open now.
Yes, you're audible. You're audible, yes.
I'm audible now.
Yes.
I'm audible now.
Yes, sir. Please go ahead.
Okay, and this I have to share, no, like this presentation. I have to share this one. It is visible?
Yes, sir. It is visible. You can make it.
Okay.
In the,
Anyway, yeah, thank you very much. I'm starting. Yeah. So first and foremost, the principal of the college, Dr. Kalal, the vice principal, Dr. Manjusha, and the convener of the program, Dr. Sudesh Goderav. Thank you for organizing this wonderful webinar, and so timely webinar. And recently, just we had a wonderful talk by Professor Nathu, and he spoke really very nicely and some of the very new things. And what today I'm going to speak, as per my talk with Sudesh Goderav, some chemical education kind of thing, to be spoken with the participants, and particularly those who are teaching undergraduate and postgraduate classes. So I was thinking, what to talk, and then I decided for the first time, I think, let us speak about the chemical revolution by Lavoisier. And I will just let you know why I chose this particular topic, and about because we all have heard about the French Revolution, we have heard about American Revolution, but what is this, the chemical revolution? But this is something very important. This event has taken place parallel to, it is contemporary to both the things which I have spoken, that is the French Revolution and the American Revolution. And whatever has happened, we need to convey to our students in the classroom to inspire them and to motivate them. So this is, we all know that Lavoisier has been adjudged and is well known as the father of modern chemistry. And so I'm showing, the Lavoisier with his wife. And what I'm showing, why I'm showing his wife, because this lady contributed a lot.
Dr. Parekh, can you go on the, sorry to interrupt you, can you go on the presentation mode, please?
I'm audible. Should I continue?
You're audible, but can you make your PowerPoint into a presentation mode, please? If you can see at the bottom half near the scrolling toolbar, you'll have.
Oh yes. Yeah, it is to the slide. Right. Okay. Fine.
Please make it in the slideshow.
Yeah, it is
mode,
okay.
Sir,
it is better. It's okay, fine.
Not yet, sir. Yes, now
thank you.
Thank you.
Yeah. So what I was telling I'm just showing the Lavoisier and his wife, because the wife has contributed enormously in all the outcomes of the Lavoisier. Let me just take about the purpose of my talk. This is the landmark lesson of chemistry. Something which has really significant contribution done by scientists in various times. So I just call it as a landmark lesson. And we need to just explain our students, how the scientific processes are explored. And when we tell them, what kind of difficulties the scientists face in their times, against the political setups and all kinds of social disturbances, but they have done it, so that is really enough to inspire and motivate our students. So, and the kind of tenacity they have, that the fight and the rebellion they have, and the commitment for science, and to bring out the new for the society. So that is what the whole purpose of talking to today of my lesson. So anyway, just let us start with this the cover page of a book, and that is the elementary treatise on chemistry. It was published in 1789, and it is considered to be the first modern chemical textbook. And that was the book by Lavoisier. So we need to know, what this book contains. Because although we know that Lavoisier has done so many things, let us see what the book contains. So that book has, the information about the influence of heat on chemical reactions. How the chemical reactions are affected by heating and all kind of things. The nature of gases, the composition. Then the reaction of acids and bases to form salts. This is, I'm talking about the content of that book. The apparatus used to perform chemical experiments. This is very important, because those in 1789, it was not they didn't have sophisticated instruments, and they themselves, designed their own apparatus, and thereby just finding out their inquisitiveness showing that. So then this book contains, for the first time, about the law of the conservation of mass, and Lavoisier asserted that in every operation, an equal quantity of matter exists both before and after the operation. So anyway, the law of conservation of mass is a part of a smaller class of the science and chemistry book these years, but it was something very, very important in those days. And perhaps, this was the first time the table of the simple substances, the classification of the elements was done by Lavoisier, and that also is a part of this book. So just I'm showing, that the book, written by Lavoisier, is considered to be the first modern chemical textbook contains all these kind of things. So let me just tell you about the Lavoisier, how he became a scientist, what was the societal status of France from where he was, and how he fought all those kind of things. So like in any other house, the outcome, that was prevalent in the society. And but Lavoisier was there to dispel the clouds, and to bring the real science out of his experiments. The chemical heritage of Lavoisier. See, he was hardly 17 when he was in a college in Paris, and in that time, the chemistry was really not considered as a true science. Just we have to go try to understand that. Of course, physics, by that time had come to age because of the work of Isaac Newton a century earlier. And chemistry was really mired, it was under the cloud in the legacy of the Greek philosophers. Those, like the Aristotle, who used to say, I will show you, this was, somewhere around 400 BC, that is almost 2005 years ago. And what was the concept prevailing that time by the Greek philosophers? That the whole creation, the whole world is made up of earth, water, air, and fire. So that means the air was considered to be as one of the elements. And it was Joseph Priestley and Lavoisier, together that dethroned an idea that dominated science for 23 uninterrupted centuries. It's a very, very long time. 2,300 years. Everybody was of the view that air was an element. But we all know now that it is the Priestley and the Lavoisier what they have done. So let us see how they have done and what kind of things they faced. Concept of phlogiston. Thrown into the mix was the concept of phlogiston in those days. And that was developed by a German scientist, Stahl. So Stahl believed that every combustible substance contained a universal component of fire, which he named phlogiston, from the Greek word inflammable. So that was the theory of phlogiston. Everybody was knowing that anything which is combustible must contain a substance known as phlogiston. Then only, and because of that only, it burns. So that was the theory given by Stahl. And because the combustible substance like charcoal lost weight when it burned, Stahl reasoned that this change was due to the loss of its phlogiston component of the air. So when a charcoal is heated, when it burns, and in the end you get an ash which has a lower weight as compared to the initial. The charcoal was taken. It is because the phlogiston, in the process of burning, goes away, and therefore you are getting a substance which has got a lesser weight. So that was the phlogiston theory prevalent. And today we understand, that really is not the case. But how, the Priestley and Lavoisier kind of people, they just dispel those kind of information and those kind of theory. Turning from organic substances to metals, it was further suggested that less residue, a substance left on burning, the greater its phlogiston content. That was the thing, that anything is combustible having phlogiston, and if suppose you are getting a lesser residue after burning, that means the substance earlier you were burning having more phlogiston. So now turning from the organic substance like charcoal, the scientists of that time, they said about the metals. The Stahl try for the metals also. Stahl knew that a metal cult heated with charcoal formed the original metal. This metal quartz means quartz, they call it the oxide. So metal oxide, when heated with charcoal, it gives metal. That was known that time. So this star proposed that the phlogiston of the charcoal had united with the quartz when it is, burned, and you are getting a metal. Now, what was the thing? And therefore, the metal which were thought to contain phlogiston were also classified as combustible. Now that time, they say the metal was also combustible. Now, the problem was this, the reverse reaction. The difficulty with this scheme was the reverse reaction. When metals were strongly heated in air, the resulting quartz weighed more than the original metal, not less, as would be expected if the lead had lost the phlogiston, come more weight. So that was not explainable in those days, and that was really could be a setback for the theory of phlogiston. But there was nobody up to that time, who could challenge this theory, and it was later the Lavoisier. Study of combustion. So this inconsistency, which inconsistency? The phlogiston going out, getting a substance and residue of a lower weight side, it is fine. But when you are burning something, but still, you are getting a substance with higher side weight. So that was the inconsistency. So this inconsistency caused some phlogistists to suggest that phlogiston might have even a negative weight, something very strange. Lavoisier was introduced to phlogiston when he was in the law degree. They see we need to tell our students, like a scientist, when he's taught one particular theory, one particular concept, he grows with that. But when he is an intelligent guy, when he is a committed person, some critical thinker, he's there, he challenges that particular theory and concepts and come out with more reasonable and more logical things. So that is why I have written it, that Lavoisier, like any other scientist, he was taught phlogiston theory. Somewhere in 1772, having abandoned law, that is what we have seen, that he has left and he pursued science. Now, combustion and the attack on phlogiston. Lavoisier really, he was working on combustion. He wanted to understand what happens when something burns, and he was really trying to attack phlogiston theory. In experiments with phosphorus and sulfur, both of which burned readily, Lavoisier showed that they gained weight by combining with air. With lead quartz, he was able to capture a large amount of air that was liberated when the quartz was heated. So that was these were the two things. Sulfur and phosphorus got more weight, but when the lead quartz, that is an oxide, was burned, he collected a gas, and that we know all about what was that gas. So to a suspicious Lavoisier, these results were not explained by phlogiston. So now almost he is on the verge of attacking the phlogiston theory, and all because of his, he was a very good experimentalist. He was carrying out all small but beautiful experiments utilizing apparatus of his own, designed by his own. Now the combustion actually involved air. Although Lavoisier now realized that combustion actually involved air, the exact composition of air at that time was not clearly understood. That is there. So now here comes a kind of a collaboration. How the scientists, they collaborate the information from different scientists. They collaborate and then come out to a better outcome. So here, on a parallel side, Lavoisier was in France and Joseph Priestley, he was in England, and he was working on all the cases and air. So 1774, I'm showing, he may not be the Priestley, just a picture, So this experiment, is a very simple experiment, but very important experiment. Let us see. And I always tell my students about the Priestley who discovered oxygen in 1774. I asked him, what was there, before 1774 when the Priestley first discovered oxygen? So some of the younger students, they really think, but anyway, they realized, that there was an oxygen, but it was not, isolated, or it was not identified by the time. So anyway, so that is the thing. So just by using a 12-inch wide glass lens, Priestley focused sunlight on a lump of mercuric oxide in an inverted glass container placed in a pool of mercury. Pneumatic, these guys, those scientists, those who were working with the kinds of airs, they were just called as pneumatic. So this small experiment where he is just decomposing mercuric oxide and thereby collecting a gas. The colorless, odorless, and tasteless gas emitted from the lump caused a flame to burn intensely and kept a mouse alive about 4 times as long as a similar quantity. I mean, this is in continuation to the Priestley's earlier experiment, that he was doing with the mouse and all, and the mouse just dying and all that. So with this particular, air, they used to call it air even in that time, or we can call it a gas. 2 things happened, that the flame burned intensely and the mouse, live longer. So we understand, now at this time, that in fact they were talking about oxygen, and these are the properties of oxygen. So anyway, Joseph Priestley, I was telling you, and then this Joseph Priestley, somewhere in 1774, he visited Paris, from England to Paris. And when he went to Paris, he met Lavoisier, and who was working on quite similar thing. And then he described how he had recently heated mercuric carbs, that is the oxide, and collected a gas. The whole experiment he conveyed to Lavoisier, and on this strong foundation and this information, Lavoisier really later came out with the idea that what the Priestley's air, in fact, that is an oxygen, dephlogisticated air. Now, see how they were struggling. Even up to this time, Priestley or Lavoisier, nobody was using the word gas, nor they are using the oxygen, nothing. Priestley believed his means the gas he got just by decomposing the mercuric oxide, pure air, I call it pure air, enhance respiration and cause candles to burn longer because it was free of phlogiston. See the cause. What the Priestley was of the view, even up to this time, that if suppose the pure air supporting the burning of the flame, it is because it was free of phlogiston. So that shows that the concept of phlogiston was still there. And for this reason, he called the gas that he obtained from decomposing mercuric core as dephlogisticated air. That's all. The name of the gas, it was not named oxygen because we know that it was named oxygen by Lavoisier, but the Priestley called it dephlogisticated air. Now, in Paris, Lavoisier repeated the same Priestley's experiment with mercury and other metal gases. He concluded that common air, now the word is a common air. What he's saying, Priestley was saying it, pure air. Common air is not a simple substance. No, that is the different. Common air is the normal air. It is not a simple substance. Instead, there were 2 components. One that combined with the metal and supported respiration, and the other an asphyxiant that did not support either combustion or respiration. So that is what the idea of Lavoisier. 1777, Lavoisier was ready to propose a new theory of combustion that excluded phlogiston. Now, up to this stage, Lavoisier was convinced, and after having met Priestley and seen all these experiments, that this phlogiston theory is not going to stand longer. He said combustion was the reaction of metal or an organic substance with that part of common air he termed eminently respirable. So by this time, Lavoisier could tell them to the scientific society, that common air that we call air, has got more than one component. One supports burning and one does not support. So now he said, 2 years later, he announced to the Royal Academy of Sciences in Paris that he found that the most acids contained this eminently respirable air. Therefore, was calling it oxygen, from the 2 Greek words for acid generator. So now the first time this gas, or the air, with the part of the common air which supports burning, which supports respiration, was called oxygen by Lavoisier. The full-scale attack on phlogiston. Now he was ready. He was of the view, and then Lavoisier began his full-scale attack on phlogiston in 1783, claiming that stale phlogiston is imaginary, because really it was not answering many of the questions. Now, calling phlogiston a veritable Proteus that changes it from every instant. I mean, every time you are giving, because at one time, those who are in the favor of phlogiston, they said there is a negative weight and all those kind of things, not very reasonable and logical. So this is the theory of combustion. The Lavoisier asserted that, he said, to distinguish what is fact and observation from what is system and hypothesis. I mean, he wanted to attack on, something which is very hypothetical kind of thing. And he was doing all experiments, and on the basis of the various analysis, on the basis of the series of analysis, he deduced the things and validated things. So that was the whole thing. And now phlogiston sustained, the idea of phlogiston sustained very long, more than 100 years like that. And so Lavoisier, when he came out with this idea that there is nothing like phlogiston and only it is the one part of the air, it supports the burning. So Lavoisier was not expecting, like this particular idea will be adopted soon. So he says, they adopt new ideas only with difficulty. Because that was a very old idea of phlogiston. So Lavoisier put his faith in the younger generation who would be more open to new concepts. And he was not disappointed, because later on it was accepted. One more thing, in a parallel way, a new chemistry emerges. Somewhere in 1776, Cavendish isolated a gas that is, he called inflammable air, because it burned readily. We know that is hydrogen, but in that time it was called inflammable air. And for Lavoisier, combustion meant combining with oxygen. So until he could explain the combustion of inflammable air, he was of the view that people will doubt this finding. And Priestley noted that when inflammable air and common air were ignited with a spark in a closed vessel, a small amount of dew formed on the glass walls. Inflammable air, that is hydrogen, and common air that contains oxygen, so that forms H2O, that is dew. 1783, Lavoisier learned from the Cavendish experiment and immediately reacted oxygen with the inflammable air, obtaining water in a very pure state. He correctly concluded now that water was not an element, but a compound of oxygen and inflammable air, that is hydrogen we call these days. So that was very important finding from Lavoisier, that he said the water is not an element, it's a compound of hydrogen and oxygen. And to support his claim, Lavoisier decomposed water into oxygen. The both way he has done. He formed water, the dew, and he just tried to decompose water into oxygen, and both ways he has done it. So when Cavendish repeated the experiment, he found that the dew was actually water, and the Cavendish explained the result in terms of phlogiston. Again, he was in phlogiston kind of thing, and assumed the water was present in each of the two airs before. See the thing. When phlogiston was so much in their mind that Cavendish was trying to explain the formation of water, that both the gases, they are having water in both the airs having that particular kind of water into it. So that was something very strong. Here it is something very important. Then I will just take 5 minutes. The last objection to discarding phlogiston now could be eliminated. It was known that when a metal dissolved in an aqueous acid solution, it produced a salt and inflammable air. That these days we know, when you put something in an acid solution like a metal, you get salt. You get a hydrogen. So metal plus acid plus water gives salt and inflammable air, which Cavendish believes phlogiston in those times. Again, Cavendish was there only. Then metal, that is the calx. Calx means the metal oxide plus phlogiston, plus acid, that gives salt and inflammable air. Okay, so this was the whole thing, and by this time, Lavoisier has really done about his theory, the phlogiston theory was removed because of him and because of Priestley, and he just came out with very good ideas and all new theories. In 1791, mostly the young scientists and chemists of those time, they accepted his ideas, and it lasted for 200 years later on. And I just, in the end, I talk to my students, the Cavendish, the Lavoisier and Priestley, together they have done so much of good. Even Lavoisier, considered as the father of the modern chemistry, he has contributed enormously in so many fields, as I shown, in the content of the book. But this fellow, Lavoisier, was gelatinized. It was a time of French Revolution. The American Revolution was also taking place. And the word chemical revolution is coined by Lavoisier himself. I asked them, to the students, probably today, you are all, you are not the students, if the students are there, I may ask them, that can you imagine, what would have been the reason that a scientist, a chemist of that stature, who has contributed enormously in the field of chemistry and science, he was gelatinized. Gelatinized means there was a guillotine, where the heads are put and they are chopped off immediately. So what could be the reason? So, the students try to, sometimes, they say, because they know, the French Revolution was going on, so they thought he must be against the king of France, the Louis of that time and all this kind of thing. But it was not like that, the story goes like that, he got money from his mother's side, and in quite early years, of his work, he bought a company, a company that is a firm general, so he got a share from the firm general, and that company was collecting different types of taxes from the common man on behalf of the king of the France, and the people got against him, and he was all the members of the firm general, that firm, which was collecting the taxes from the common men, they were taken into custody, and it was a reign of terror, and he was tried, very formally, and he was guillotinized. He was killed, his head was chopped off. So probably he would have never thought, while just purchasing the share in that company, but it happens in the life anyway. And his friend, that is, we all know, the mathematician, the Lagrange, he remarked, it took them means that people, they chopped off, only an instant to cut off his head, and 100 years may not produce another like him, like Lavoisier. So thank you very much. And I think this is a time for me to take a cup of tea, and this is the years of webinar. So thank you very much. Thank you. So nice of you. Thank you, sir. I have spoken.
Yes, thank you, madam. Thank you, madam. मराठी मैडम अनम्यूट करा. Madam, unmute yourself. Madam, unmute yourself. Rathi madam, unmute yourself. You are not audible, madam. Unmute yourself.
Hello. Am I audible now? Okay, thank you.
Yes.
Professor Brijesh Pare, I want to say one thing. I really liked the title of your presentation, Lavoisier, a Child of the Enlightenment. It was really wonderful title. And one more thing I want to tell you, sir, your chemistry with the history of Lavoisier was outstanding. And your session was really full of passion, and you gave the priceless knowledge on some of the unknown facts. Now I am also in love with Lavoisier history, and I also want to read more the way you have explained. Thank you, sir, for explaining the landmarks in the French and American Revolution of chemistry. We have, as a chemistry teacher, we have never really gone in the detail about the these details. Thank you so much for making aware of all these things. So once again, it was really very wonderful sessions to hear you here. Thank you so much, sir.
Thank you so much. And I take this opportunity to thank Dr. Sudesh Bhaskar, the convener, Khadera, principal, and Professor D.V. Prabhu, the General Secretary of SCT. He was here. I'm thankful to all of you. Thank you.
Thank you, sir.
Thank you so much. Now, dear participants, we are proceeding towards our next and the last session, the third technical session. So, is it a break or we are directly starting, sir? Godera, sir?
We are directly starting, ma'am. We have limited time.
Okay.
Yes, sir.
So now I'm handling this session to SCUBE representatives.
Hello. Hello.
Charitra, you're audible. You can continue the session.
Okay. Thank you. Thank you very much to all the panelists, Dr. Sudesh, Dr. Nathu, Dr. Meenakshi, Dr. Manjusha, Dr. Brijesh, Dr. Kalal, and also thanks to Mr. Subhash to be a part of this this webinar. I hope in the few next minutes, all the students and the faculties and all the research scholars will having the insight of the features and the functionality of the ChemDraw software. And I hope all can see my screen, right?
Yes, sir.
Perfect. So now we will discuss about the ChemDraw. The latest version of the ChemDraw is 19.1, right? So we all know about the ChemDraw, right? It is the part of chemical drawing tool, very famous chemical drawing tool. And apart from the drawing, it can do the prediction, the reaction prediction, the analysis, the calculations, right? It can give you the various supported by the various file formats, right? So in this session, we first discuss about the presentation part, which we will cover the insight of the ChemDraw, and then we will present the demonstration for ChemDraw, right, with various examples that I have already prepared for this particular webinar. So let's begin. Firstly, let me introduce myself. My name is Charitra. I have done my master's in pharmaceutical chemistry, and I'm working as a technical consultant in SCUBE Scientific Software Solutions more than 5 years. So I'm just handling this computational chemistry part along with Dr. Saurabh Sinha, who is my colleague. Let me begin with the outline. So we will cover the ChemDraw templates. So there are predefined templates in the ChemDraw, biological templates, structural templates, right? Some biopolymers are there. So these are the predefined templates that are saved in the ChemDraw. Then we have drawing with hotkeys. So instead of using your mouse, you can just write down your structure with the hotkeys. Name to structure, name generation is there. Generic structures, bracket expansion, rings color filling. This is the new feature in the ChemDraw 19. Copy paste from Smiles, Inkai, Helm, TLC, gel electrophoresis, stoichiometry calculation, Helm toolbar, some defining dictionaries, prediction NMR, add variable attachments, searching with the ChemACX, PubChem GHS LCSS. So lab chemical safety summary is there in the PubChem. And then we will cover the Chem 3D part. So how we can do the conformational analysis with the Chem 3D that we will cover. Right. So ChemDraw is a drawing tool since 1985. ChemDraw stands for publication grade quality. So there are certain formats, general formats are there like GMatChem, GNatural Product, RCS, right? So all these are there in the ChemDraw itself. So every journal having their own format for the drawing. So that is all there in the ChemDraw. User friendliness, as I have told you, hotkeys. With the help of hotkeys, you can draw the molecule. Chemical intelligence. So ChemDraw is the chemical ear and brain of PerkinElmer Informatics. So it's a popular product of the PerkinElmer. Hotkeys. So if I press 1, it will be single bond. If I press 2, it would be double bond. If I press 3, it would be triple bond, right? Likewise, wedge bond, hash bond, partial bond, that all can be drawn with the hotkeys. Not only bonds, but also the atoms we can draw with our keyboards. So in this way, you can draw a molecule 3 to 5 times more faster. So as I've told you, this is the hotkeys, their particular numbers and their particular letters, which is also customizable. So you can customize these hotkeys and do your research part, do the drawing part, right? So it would be very easy to draw. Instead of mouse, you can just use your keyboard. So if you can see here, if I press Z, capital Z, small z, O, N, so there are certain functional group can be added by just press some keys from your keyboard, right? Helm toolbar. So if you can see, the Helm is the hierarchical editing language for macromolecules. So it's basically a format becoming the industrial standard representation. So if we have a larger kind of bulky DNA fragment or RNA fragment, then we can, in a short form or in a text form, we can convert it into a Helm, and Helm is supported by ChemDraw, and then we can get the structure, right? So that all we will cover in the demo session. So the Helm toolbar is basically in a package of the ChemDraw Professional and Chem Office Professional. So there are 3 packages we will discuss in the later part of this presentation. Then we have a ring coloring option. So ChemDraw Professional and Chem Office Professional, both packages are having this ring coloring option. So earlier, this is not there in the ChemDraw, but on the basis of all the queries of the scientists and the users, prestigious users of the ChemDraw, now PerkinElmer has added the ring coloring option, which is very useful for the researchers. Integration with SciFinder N. So there is a SciFinder and SciFinder N. Both are integrated in the ChemDraw. So if you have an access of SciFinder, then you can search your structures, substructures. You can search the retrosynthesis part, patent pack, some new methodologies of synthesis that can be searched by SciFinder. And lab chemical safety summary. So there are environmental hazardous chemical, there are physical hazardous chemical, there are health hazardous chemicals. So all these regulations or protocols for the safety are there in the ChemDraw. So you just draw your structure, select that particular structure, and you will get the their particular safety summary. You can also paste it into the working window, and you can get the statement directly from the pumpkin. So as we told you, there are 3 packages available on Windows and Mac. So ChemDraw Prime is there, which is known to be as you can just draw the structure, copy paste into your document. Then we have ChemDraw Professional, which includes all the features and the functionality of the ChemDraw Professional. Helm toolbar is there, Chem 3D, Chem Excel is there, ring coloring option is there, and SciFinder N. Then we have Chem Office Professional, which includes all the features and the functionality of the ChemDraw Professional. We have LCSS PubChem, interface of third-party software like Gaussian, like Autodoc, Gamers, Conflex, right? So it acts as a interface, Chem 3D. Then we have ChemDraw MNOVA Lite version, in which you can predict and analyze the 1D NMR. Then we have Signals Notebook, which is cloud-based electronic lab notebook. Now, just let me continue with the demo session. If I can show you the interface, right? So this is the interface of the ChemDraw. Now there are 37 features and the functionality that we will cover in this demo session. I have already prepared some example, which I will show you during this particular. If we will cover the particular topic, I will show you the respective file that I have saved. And also, we will cover the Chem 3D part after finishing our ChemDraw 2D part. Right? So let me begin with the templates. So if you can see here, there are the templates for the BioDraw. There are templates for amino acid chain, amino acid, anatomy, animals, aromatics, bicyclic, bioinstrument, bioart, bugs, clipware, some experimental glasswares, conformers, CP rings, right? Cycloalkanes, DNA templates, nanotubes, polyhedra, polypeptide, RN template shape, some stereocenters are there, supramolecules are there. Right? So just you can choose one of them and just click once, and you will be able to work in your window. Some of the you can also define the new templates. There is a section for the new templates right here. So let's say if I am working with the streptomycin, right? This is the another aminoglycosidic antibiotic. So let me just start with the name to structure. So and to mycin. Right. So just write down the name, and you will be able to get the structure. So there are more than 1,000 FDA approved drugs that are incorporated in the ChemDraw itself, and they are continuously updating it. I will show you in the later phase of the demo that what extent we can utilize this name to structure feature of the ChemDraw. So what I can do, suppose I'm just working with the streptomycin, and then what I can do, I want the streptomycin structure with my templates here itself. So next thing I can do is put the file, save as, and streptomycin. It is saved as .ctp file. Save it, right? And once you open the template, you can see the STP, streptomycin, right? So next time you open the ChemDraw, it will be saved as a template, and then you can directly choose it, and then, oh, okay. Then just click once, and you will be able to copy the structure. Some biological drawing is also there. So once you go to the file and go to the sample, there is an alcohol metabolism, alkyne terminal, antigen pathway, carbon hybridization, citric acid cycle, codon triplets, right, GPCR pathway, some ignite reaction, right. Then I can show you the citric acid pathway. Right. So these all are the predefined samples. So you can also edit it. Once you click on this, it will not allow to edit it, but when you just need to copy and paste to the next document, and you will be able to edit it according to your research area. If I can show you my personal files that I have created. So I have created the types of ELISA test, right? So there is a direct ELISA, there is indirect ELISA, there is sandwich ELISA, competitive ELISA. So that all you can draw from BioDraw toolbar. So there is immunomodulators, there is antigen, ion chain, G protein, enzymes, ribosome, microorganism, DNA, RNA, helix protein. So all can utilize for your particular biological drawings. Right. Now, let me show you the hotkey part. Just close this one. And when you go to the samples, magic hotkeys cheat sheets are there. So once you click on this, you will be able to get all the forms with their particular hotkeys. If I press 0, it will be single bond. Press 1, it will be single bond in the same direction. 2, keto group. 3, benzene or benzyl ring. Right. So likewise, if I press 6, it will be cyclohexane. Right. 7, cyclopentane. So likewise, you can just draw your molecule. So for example, this is the paracetamol molecule. So if I just click 2N1310, you'll be able to draw the structure in a fast way. Right. So that is there in the file, open sample, magic hotkeys cheat sheet. Now, let me start with the drawing part. So here you can see the solid bond, right? So all the toolbars are there. We just take single bond. Now, once you click on the single bond, you would be able to see the green dot, right? Which will tell you the next direction, the atom, which you would like to draw, right? So let's start with the 1, 1, 1, 1, 1, 1, 1, 1, right? And there should be NH, which is 1, 2, 4. Then there should be sulfur here, then single bond, sulfur, single bond. Then there should be 1, Z, and right? I'm not using my mouse, just the keyboard. With this keyboard, I can draw this particular structure. So this is basically a cyanoacrylamide compound. In a recent journal, I have gone through, and this is quite useful for fluorescence, and it's also having some biological activity. Likewise, if I would like to draw some bulky molecules, like there is a newly synthesized aminoglycosidic antibiotic called as the plasmicin, right? So if I go to the name to structure and this plasmicin, okay, there is some text error, right? So this is the structure of the plasmicin. Now this is the chair-like structure. So if I can draw the front perspective structure, so what I can do is just let me draw the benzene ring. Right? Select it. Now with the single bond, 1 OH, there should be the NH, then there should be O. Okay, it's 1 single bond. It should be double bond, 1, 1. Then there is NH2, there is single bond, there's O, then single bond, there is 6 for cyclohexane, then single bond OH, then we have OH here, then we have single bond O, single bond 6, right? Then we have amine here, then we have OH here, then we have OH here. Then there is a chain is there, H, CO group, H, right? Now there are some hash bond and wedge bond. So what I can do, if I press H, it would be converted as hash bond. Like H here. Then for the wedge bond, I can just press W. Whole bond, W, W, right? Now, if I can select this molecule and go to the view, object, then there is a show stereochemistry, right? So S and R conformers are there. And if you can see, there is a red circle over here. So it is what kind of warning it is showing. So this stereo center has no stereo bond specified, obviously. So what I can do is I just hash mount. There should be the hash mount. There is wedge mount. Right. Okay. So this is the front perspective of the plasmicine. In this way, you can draw the certain structure a few seconds or few minutes by using your keyboard. Suppose if I would like to draw the glucose part or Fischer projection of the glucose, for example. So what I can do is just select the single bond, right? Then 1, 3, 4, 5, right? Then 1, 1, 1, 1, 1, 1, 1, 1. So for the text part, just press T, and you would be able to type functional groups. CHO is there. Then there should be OH, there should be H, there should be H, there should be OH, OH, sorry, OH, OH, H, H, and then the next thing, just type down CH2, OH, right? So this is your Fischer projection for the glucose. And if you would like to clean your particular molecule with the stereo centers and all, so what you can do is Shift Control K, you will be able to clean your structure. Right? So multiple times you can click on Shift Control K, and you will be able to refine your structure. The next thing is the perspective drawing for the glucose. The same. So what I can do, I can just start with the cyclohexane. Select it by press space, right? Control R for rotating the angle. So I would like to rotate it 90 degrees. CW. All right, and then you can just need to increase the size. Right. Put oxygen over here. Then let me begin the single bond. Here, 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 here. So here. Right. For text part, just press T, and then you will be able to enter the functional group. CHO is there. There should be H. There should be OH. There should be H. OH. Should be OH. H. Should be, I think, H. OH. Right. So here we have a perspective drawing. Then the last thing is the Newman projection for glucose. So what you can do, just select the single bond, select 1, 1, and enter the functional group H, H, H. Now to copy the structure, select it. Then what you can do is press Control, then just drag it down and attach single bond to the particular carbon centers. Now, if I would like to add the bond between the carbon to this carbon, it is fixed in the length, right? But if I go to the particular atom, if I move my cursor to the particular atom, it will automatically attaching the bond. So fixing and unfixing the length from object, you can deselect and select from here itself. Now, let me just select this particular molecule and Control R to like 90 degrees, right? Then what we can do is I just need to select the orbital, select from here, and just select the back drawing, go to the object, and then what I can do is I just need to bring the front like this way, right? So here you have demand prediction for that. Because what else we can do is we can draw some complex molecule like a bicycle pentanes or BCP. So, so obviously it is showing some warning sign. So what you can do, you can just deselect from here, display warning sign. So in the next page, just draw. Right. Now, let me draw another molecule. Or this is basically bicycle ring. So what I can do is just begin with a single bond, then Z1, Z for triple bond basically, and then 5 for pentane. This is 1. I should delete it here. Then B for cyclopropane. Delete this one. Now, if I choose the bond between this atom to this atom, it will automatically attach this atom to this one. Right? You can see the front bond is this one. Now, if I want to create this bond as a front bond, then just press F, and you'll be able to get the front bond like this. So for wedge bond, like this. Right. So just keep it here. Select the benzene ring. Control R. It should be 90 degrees. And this would be single bond. And text to C3. Now, here comes the trick that if you would like to add this atom to this one, so what you can do is you just select it, keep press shift, select this one, and control J. Right? Similarly, select this part, control for the copy, and shift this part plus this part, control J. Right? So this way you can draw certain bicycles. Okay. So as we are proceeding, let me just show you another example for generic structure bracket expansion. Delete it. If I have a benzene ring, sorry, okay, and I can add some bond here. I would like to have Bok. Just press Y for Bok, and then there should be O. Now for bracket expansion, if I choose bracket and the repetition unit as average, I would like to go with 2 to 5. Now select it, the whole molecule. Let's go to the molecule, molecular weight. So it will be 340.42. Now what I can do is I just need to copy this. So copy this here, and if I can change the expansion to 14, and just select this molecule, the molecular weight is changed, 655.69. Right. Now here comes another trick, that if I can expand this structure with the 14 repetition unit with this area, then what we can do is go to the atom, expand label. Sorry for this, I think it should be general structure. Right. So in this way you can draw, if you are working with the expansion unit, so just define the particular number of the expansions and you would be able to get the structure. One of the best example is for drawing the proteins or peptide. Like if I can protein having the NH and C carboxyl group. So it should be the N, it should be the single bond. Oh, right. Now if I can here to here with a repetition unit of suppose 7, then I can select it with the structure, expansion with the structure. Right? So some peptide ring you can able to draw. One of the example I have prepared for this one is PG5. So this is the largest molecule ever synthesized by the chemist, right? So here, if you can see, there's a Bok NH groups at the terminal, right? And with the repetition unit of 10,600, right? So this is the largest, you can say, dendronized polymer. Right? I can show you the actual size. Okay. And there are some properties of this molecule. Molecular mass is over 200 megatonton. Number of atoms more than 17 millions. Shape is cylindrical, diameter 10 nm. So suppose if I just expand this structure with the generic, so there is a, it will maybe, I don't know what, how many pages it will cover in the camera. Right. So this way you can draw the polymers, the peptides. Now let me show you another example with the rotation moves the center of the rotation. So Okay, let me also show you one of the example of a polymer, because we are also discussing about the repetition unit. So here we have another example. I can size. So here we have a trigger molecule. ABCD is the amino acid sequence. Right? The monomer is there. Then we have the Krebs catalyst, and we can expand it into the N number. You can see the bracket expansion with the N number. So if you would like to draw this kind of, you can see a circle. So what you can do, you just need to select the arrow, right? Then just need to add some orbitals over here, and obviously in the orbital you can draw the color, fill the color. So in this way you can draw this particular chain. All right, let me proceed with the central rotation molecule. So suppose if I have this particular molecule, if I can draw one, there should be two, O, there should be one, which, great. Now if I can select this part and I would like to rotate it, so what I can do is press Alt and the arrow. So I can select the upper arrow, it would be going to the upper side, lower, the downside. If I would like to rotate here, control, upper arrow, lower arrow, or down arrow. So in this way, I can rotate this. All right. Now moving to the catalytic cycle or eclipse cycle reaction. So what I can do is, if I want to draw some catalyst cyclic reaction, just need to select arrow. Right? So your arrow should be straight, and select it, or you can change angle to -180°, then drag the arrow part like this, and then like this. Right. Suppose if I have this benzene ring here, this benzene ring here, now what should I do to proceed with next arrow? So what I can do is just need to select the arrow, control to copy, and set it back to that arrow. Now just drag it, just drag it the tail part like this. Then some benzene here or some other molecule. This is just for example, I'm just trying. I will show you the proper example. Then just similarly copy this here, then drag it back, and then here, just drag the tail part, then just draw some scene here. Similar way, you can just copy, drag head part, then tail part. Right? So in this way, you can draw the cyclic or ecliptic reaction, and you can just draw the perspective drawings, increasing or decreasing the size. So the topic is also related to green chemistry. So I'm just would like to share one word of ibuprofen synthesis. So when I just open it, that particular file. So here you can see if I would like to share the actual size. So here you can see the ibuprofen, the old traditional synthesis of ibuprofen includes 1, 2, 3, 4, 5, 6 steps, right? This is basically not a cyclic reaction. I've just represented it as a cyclic one. So 6 steps are there in the old traditional synthesis of ibuprofen, right? But with the new green synthesis of ibuprofen, it only takes 3 steps. It's like 1, 2, and 3, right? So in this way, you can save your solvent, you can save your time, and also you can save the chemicals and their harmful effect in the environment. Right? So this 3D model can also be added in the ChemDraw. So once you go to the edit, you can get the 3D model here. Some other example, if I can show you, is the Wilson's catalyst reaction. So if I can open, right? So here you can see how the reaction is taking place. Element by color is always there. So in the color section, I can just select it. By element is there. So if you can go to the view periodic table window, so there is an indicative periodic table. So nitrogen is in blue color, carbon obviously that is in black, oxygen is in purple, sodium is in pink. So you can also, if you can just would like to customize the color, you can select from here and you can go with your drawing. Right. Now another example is toggling the aromatic display. Suppose if I get this benzene ring, this is a resonance structure, right? Now by Alt K, I can change it into the circular resonance structure. Alt K, you can see the difference, right? I can also clean anthracene, then control K. So here you can see the difference. By Alt K, you can toggle the aromatic ring. So you can go to the structure. There is toggle aromatic display. Okay, by the use of the hotkeys, I can also draw the honeycomb structure or graphene backbone. So what you can do is just select cyclohexane. Just select this one and press 6. Select the particular carbon atom. Press 1. Then select the terminal carbon. 6. Select this one. 1. This one. 6. Right. So this way you can also draw the nanotubes, the graphene, the honeycomb structure. So that is quite easy to draw. Next feature is atom by color or ring coloring option, ring color option. So select this one. Now, if I would like to add particular color for this ring, yellow. I would like to get the particular color for this one. Sorry, this one with blue, or this one or red. So this all can be facilitated in ChemDraw. There is a new feature in the ChemDraw for ring coloring option. If I can show you some pretty useful examples, let me just allow to show you. It would be very interesting. Here is heterocyclic nuclei. So, as a medicinal chemist, I would like to know about what are particular ring, heteronuclear ring is there in the drugs. So, if you can see, nitrogen containing rings having a brownish color, right? And similarly, oxygen containing rings having the blue in color, and then sulfur containing rings having green in color, right? And some other multinuclear rings having a different color. So I can show you the whole documents, right? Similar way, there are some drugs, when we take it, it will discolor our urine, right? Urine color changing drugs. So, for example, if I can show you those example, right? So, rifampicin is there, which is an antitubercular drug, which discolor the urine, which change the urine color into the red. Some nitrofurantoin is there, which convert the urine as a brown. Cimetidine, antihistaminic drug, change the color as a blue one. Bluish. Amitriptyline is there. Warfarin, red in color. Enazopyridine, brownish in color. Metronidazole, brownish in color, right? So this is what you can do with the color coding for the particular molecules, right? Now, there are some journal formats which are there in the style sheet. So, as I've told you, there are certain journal formats having certain journal formats having their own particular parameters or you can say guidelines to draw the structure. So, by selecting like J heterochem, J modeling, RSC, right, village document. So, by selecting those particular style sheet, you would be able to get that particular structure. Like if I ACS document. Morphine. Right. So this particular format is in the ACS. Okay. Now, another thing I would like to share is you can also draw your style sheet or your own style sheet. So, like, I'm just continuously drawing this one. This is in progress. So, like adrenaline, which is discovered or invented by Jokichi Takami, phenytoin, Henrik Pills, right? So you can see their related photographs. I have already also tagged the benzene ring. So this particular thing also you can do in the ChemDraw. Right? So I'm just working on it. Just continuously doing the searching with the inventor of the drugs, and I'm just writing over here. So this way you can also do. You can just draw the benzene ring, select it, then you can copy and paste the particular inventor's photo or any JPG image or TIF image or any image format. You just copy and paste it from here, and then you can send it to front and send it to back, like here. Right. Then you will be able to draw this kind of structure. Another thing is the ChemACX. I would like to show you. So, okay, let me close this one. So, ChemACX Explorer is there. So if you have any CAS registry number of the particular molecule which you would like to draw, then you can get it from ChemDraw add-in, ChemACX. So like there's 5808. Let me see, I think it should be caffeine, right? Similarly, if I can go with the registry number of another molecule like 30501, can retrieve the structure. So this is, I think, chrominone ring. So in this way, you can also get the structure with the help of ChemACX. Okay. Okay, let me just style sheet with the new document. Just close all this thing, so it should not be hampered. Okay. Copy paste option are also available. So, for example, let me just go to the Word file. So, copy from Helm, copy from Smiles, copy from Inkai, a mold text form. So, FASTA peptides, FASTA DNA, RNA. Would like to, if you would like to get the structure, you can just copy it. Right? Go to the ChemDraw, Control V or paste. You would get the structure directly. Right? Okay. Similarly, for the Inkai, just copy it, go to the ChemDraw, paste it. Right? What you can do is just copy you can paste it. It will be showing you the peptide change. Now, to get the structure, what you can do is just right click, go to the atom, expand label. Then you would be able to get the structure of whole protein. Right. So if I can show you the Helm toolbar, so view, show Helm. Like if I would like to get the structure of alanine, asparagine, isoleucine, histidine, glycine. Now what I can do is I just select it, right click, go to the atom, expand label. So in this way you can just draw the particular molecule, particular peptides. Okay. So cyclic peptides can also be drawn, like cone bridge, disulfide bridge, all can be done. So for example, like the cyclic peptide, like like for example, just select it. Now what you can do is just attach solid bond, right, from this residue to this one, and then select it, atom, same thing, expand label, and you can see there is cyclic peptide. Right. If I can show you the example, when you go to the open, let me open that to DNA, obviously, right? So AGTC, right? 5 to 3, 3 to 5 orientation, size. So I have not drawn this, just go to the Helm toolbar, to the RNA, just select adenine, cytosine, guanine, thymine, uracil, and then you would be able to just expand that structure. You can attach these partial bonds. Right. Now, another thing is the stoichiometric calculation. So what I can do is to name structure. For example, right. So here, if I have the aspirin molecule, then there is a fragmentation tool for retrosynthesis part, synthesis part, right? So let me just begin with the synthesis part. If I would like to fragment from here, right? So here we have, we can modify salicylic acid. It should be OH here, and it should be OH here, single bond, O here. Right. So we have salicylic acid, we have acetic anhydride, and aspirin, right? Now, if I move my cursor to the arrow, it will show you the red box and the pink box, which will show you like this is your reactant and this one is your product. If I click once in the arrow, it will select all the reaction. Then just go to the structure, analyze stoichiochemistry. So suppose if I have a sample of salicylic acid like 3 grams, right? So it automatically calculates the sample mass for the acetic anhydride and expected mass. Now, suppose if our density is 1.4, I think so. and the density of acetic anhydride is I think 1.06 something, and the measure mass. Let's take. I got the measure mass for 2 grams, so it will be calculating your percentage yield automatically. Right? You just need to fill up sample masses and measure mass and their particular densities, and you'll be able to get the percentage yield. Okay, another faster way to draw the structure is add variable attachments. Right? So what I can do is, suppose I would like to draw nitrobenzene, phenol, toluene, or some aniline, right? So what I can do, I just need to have a benzene ring, right? Now select it, go to the structure, add variable attachments. So once you add variable attachments, just click the single bond over here and T for text, just click once. In the bracket, you would like to add whatever functional group you would like to add. You just type it down. NO2, I would like to have OH, then we should have CH3, then I should have aniline, NH2, right? Bracket close, then select it, go to the structure, expand generic structure. Right? So here you can see all the molecules are there. I can do this structure. Right? So if your parent molecule is single, just add the functional group in the back bracket, then expand the structure, and then you would be like to get the those particular structures. Some examples I have prepared, like when you go to open, there are R groups. So like here we have a gentamicin. So gentamicin is the combination of gentamicin C1, C1A, C2, C2A, and their respective functional groups are there, right? So R group can be also be the one of the easiest way to get the repetition structure with R group. And another thing is this way, right? You can see here. So in the R1, R2, R3, I have created a group in the brackets, and then if I go to the structure, expand generic structures, so it will show you the 8 structures with all the isomers. So you can choose your structure and then go to then proceed with your works. Similarly, if I can show you the tetracycline, right? So this is the tetracycline, and there is chlorotetracycline, demoxycycline, doxycycline, minocycline, oxytetracycline, tetracycline. So what I can do is just select it, go to the structure, expand generic structure, and you will get the 36 structure with all the isomers and the premix. Right. Okay. And similarly, I have a tricyclic antidepressant. I have a penicillin. Right. So if you can work in with the R group, so just select all this component, do the structure, expand generic structure. So only 3 structure would be taken. So it will not show you the repeated structure. So if you can see, there are 2 R groups are there which are same. this one and this one. Penicillin G and penicillin B, which should not be there, but by mistake I have, or you can say deliberately I have do this because I would like to show you that it is not showing you the repeated structure. Right. Another example is the prediction of the proton NMR in C13 NMR. So let me just go to the furosemide. Furosemide is the diuretic drug. So if I go to the structure, then we have a predict 1H NMR and 13C NMR. So here you can see NMR part. So solvent is DMSO and frequency is 300 megahertz. So if you go to the preferences and Chem NMR, so the two solvents choice are there, CDCl3 and DMSO, and you can just change the frequency and apply. You will get the structure and their particular protocols for the CNMR. Okay. One of the difference between the frequency, if I can show you, I prepared the example. Right, so here you can see for this NH proton 7.81, you can see this particular peak with 500 megahertz. So with 500 megahertz, estimation is good. With the lower frequency, with 300 megahertz, the estimation is rough. So you can see here NH 10.1. So this is same solvent is DMSO, and for 500 megahertz, solvent is CTCl3. Okay. So there is some chemical properties. There is some analysis part. So like say methotrexate is anti-cancer drug. So I would like to have some chemical properties of this molecule. So go to the view, chemical properties window. Right. So boiling point, melting point, critical temperature, critical volume, Gibbs free energy, topological surface area, CMR, log S, pK value. So all these chemical properties are there. Right. For the analysis part, the zone, betamethasone is corticosteroid. The view, there is an analysis window, the formula, exact mass, molecular weight, MS to Z ratio, elemental analysis, and their predictive, you can see the mass spectrum. That's a predictive one. So you can always trace it down with the MS to Z ratios. Okay. Add numbering, add charge. So when you go to the, I have some sample, this one, OH. Now I would like to have a IUPAC name. So what you can do is you can go with the file. There's preferences. In the building and display section, there is a display IUPAC atom name on structure to name to structure. Click OK. Okay. Let me. Circles, right? So what I can do is, if I have this chain tool, and I just this one and add the single bond here to here. Select it, Shift Control K for clean, and to convert it into the cyclic to alkyne ring, what I can do is just press 3 for triple bond, select it, and then I can clean the structure, Shift Control K, right. Just need to convert it to the single bond, select it, press 1 to add the single bond to all the carbon atoms. Similarly, select it to 3 to add some benzene circuits. One of the example that I have prepared is the benzene circuit on the repetition form. So you can see. So here there is a core molecule, right? So this is benzene attached with the 3 unit of benzene, and this 3 benzene attaching with the 9, then this 9 attaching with the 27, and this 27 attaching with the 81. Right. So in this way you can draw the tetramers, neurons, right? The repetition units, alkyne circle reactions. That is all covered in a ChemDraw. The last thing is defining the dictionary. Just let me close this. Let's take an example of COVID drug, right? So remdesivir is a quite famous drug nowadays. So if I go to the name to structure and type remdesivir, it is not showing you the structure, right? So what you can do is you can define the dictionary. So you just draw the structure and define the dictionary, and next time you write down that particular molecule or the structure, it will show you that particular structure. So remdesivir is adenosine monophosphate derivative. So let me go with the adenosine monophosphate, right? Okay, because I have turned on the IUPAC name, so just okay. So this is the AMP. Now what I can do is I just need to convert the structure into the remdesivir. So it should be, I think, cyano and in this OH it should be 1. This should be benzene. For this OH it should be 1. And so I go here. O 1, 2 oxygen, right? So now what you can do is you can just go to the structure, add structure to dictionary. Oh, okay. I would like to replace it. Oh, Now, once you have defined the structure, then go to the name to the structure. I think there is a problem. Right. So here we have another form of remdesivir. So once you draw the structure, just go to the defining the dictionary and you will be able to get the structure. Now, last thing, let me go to the Chem 3D part. So we are running short of time, so let me just give you the overview of Chem 3D. Okay. So obviously you can draw the structure from ChemDraw link. So let's say this is a single bond, this is a single bond. So simultaneously you will get the 3D part. Right. Now the another way to draw the molecule is what you can do is this one. Text to structure, right? So once you get this feature, you can just draw the structure. So 1, 2. See, right? Can rotate it. So here you can see this is the trans structure. So if I would like to convert it into cis structure, so here we have a move object part. I can just here, 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 here. Then select all. So here you can see trans converted to the cis one. So very popular example for Chem 3D is the analysis for the conformation. So eclipsed one and staggered one, the ethane. Right? So for example, let me just go to the text chain, right? And you can see here, I would like to generate all the dihedral angles. Suppose if I choose this one, this is minus 60 degree around, right? Now, if I would like to convert it to the 0 degree, right? It is, I think, eclipse conformer. And then if I calculate the energy, so I would like to have a pi bond steric energy and steric energy details. Just run it. So total energy of this frame is 4.143 kilocalorie per mole. Right? So this is the staggered one. Now, if I change sorry, this one, I think it's eclipsed one. If I change with the 60 degree, this dihedral angle, and then if I calculate properties, run. Now the total energy of this frame is 1.143. So steric energy of eclipsed one or 0 degree conformer of ethane and staggered or 60 degree is more likely to exist. If I can generate a single angle plot, right? So the lowest energy is having staggered one, and the highest energy is having ecliptic one. Just move my cursor to the particular area of the or particular point of the graph. It will show you the corresponding structure. Similar way, what you can do is you can do the MM2 calculation, right? MM2 minimization, right? So here you can see the stretch, the bend energy, stretch energy, torsion, van der Waals, total energy. So minimization can be done. Another feature is the interface. So you can see there are interfaces of the complex, gamers, Gaussian, MOPAC, Hotdog, right? So directly just draw your structure, and if you have these software installed in your system, then you can directly set up the job. Like this is the Gaussian interface. I can do the minimization, predict IR, NMR, UV, run frequency calculations. Right? So all these things can be features and the functionality are there in the Chem3D software. Another thing is the stereochemistry part. So if you can so let me go with the so epinephrine is a stereo center drug which is having a stereo center. So if I can go to the structure, there is a detect stereochemistry. So C5 and C5. Okay. C5 is C5. So the labels. Okay. So this is the C5. This is the stereo center in which OH group is attached. Right. Similarly, if I select this particular bond and then calculate the dihedral angle, single plot. So here you can see the conformation and their particular energy. So lowest energy having the stable structure. The highest energy is the unstable structure. All right. So there are a lot more features. You can go through the help section. There is always a tutorials, not only for the Chem3D, for the ChemDraw also. If I can go to the open, what are the file formats that are supported? So there are a bunch of the file formats: mol, .mol file, SDF file, protein data bank, PDB file. This is the interface for the Gaussian. So GGF file, input file for Gaussian, gamers, MOPAC. Right? So all this thing is there in the Chem3D. All right. So if you have any question, then just let me know. You can just drop a mail if you have any query, or you can drop your question to the question box. So if I can answer your queries.
We have a couple of questions from people here. One is from Tushar. It says, can we get point group of any molecule?
Point group?
Yes.
I don't think so. Point group is there. I need to check and get back to them. I don't think so.
Tushar, we'll email back to you once we get a clarity on the question. We'll email back to you on the answer. Another question is for mirror plane and axis. Wherever we need no command in JMOL. That command are difficult, most use, but that's more of a statement than a question. Let me check if there are more questions later to ChemDraw. Can we put our own axis of rotation other than default XYZ, which is passing through center of gravity?
In Chem3D, you can do this. Yeah, so if you have their mail ID, I will share one document so that they can do this.
Perfect. I'll do that, Charitra.
Yeah.
All right. Thank you, attendees. Thank you, Charitra. Thank you, panelists, for providing us this opportunity to showcase the greener aspects of drug discovery and the features of ChemDraw. Any sort of feedback or any sort of questions that you have, please let us know on our emails. We'll be happy to address those questions. Please fill the feedback forms, and also you will get a link of the feedback form in the next subsequent mail. Fill those feedback, and we'll be able to assist you with further queries that you have that were missed out in the session. We will be getting that done to in the email part of it, right? Dr. Sudesh, you want to add something, or can we close the session now? Dr. Sudesh, do you have any closing statements, or we can close the session?
Hello. Yeah. I think there are no questions, and if further any questions are there, of course, you have said that you are able to reply through the mails. The last part of the webinar is just a formal Thanksgiving. So I must thank Charitra Gaur for giving this informative session on the ChemDraw. And as far as this webinar is concerned, I must thank all the persons, institutes who are associated with organizing this successful seminar. One of the important one is Dr. Ram Kulkarni, who is the Zonal Secretary of Vocalization Society, the Principal of our college, Dr. Dhanesh Kalal sir, Professor Dr. Devi Prabhu of the Association of Chemistry Teachers, Professor Nathu of ISER Pune, Professor Brijesh Paren, the President of ACT. They have given their valuable thoughts, their knowledge, they spare their time. So I must thank on behalf of the organizers to this. Thanks are also to Dr. Saurabh Sinha, Charitra Gaur for sharing their expertise. And important, Subhash Kumar, who is from the Perkin Elmer, for hosting and cooperating throughout this right from the beginning for this successful of this seminar. Thanks are also to Mr. Devashish, Aninde Ghoshal and Mahendra from the Scoop team for their technical support and other relevant support also. I must thank Dr. Manjusha Kulkarni, Vice Principal and IQA Coordinator of our college, Dr. Meenakshi Rathi for anchoring this session. And I must thank Dr. Sanjeevan Kharath, HOD of Chemistry Department of our college, the organizing committee members of this webinar. And at the last, I must thank the attendee of all these webinar for their patient listening and for their cooperation. Hope in the future we may extend such kind of the cooperation also in future, and we may have such kind of webinars to be organized in near future. So I must thank all those are involved and those who have attended this seminar. As far as the feedback form is concerned, it is available in the PDF form in the handout section. Just download that feedback form. It is also given in the chat box. Also, the link is also given, so you can copy paste in the URL. So you can fill the feedback form, and in a 2 days of time. So after filling the feedback form, the certificate will be issued to you in a period of weeks time. So thank you one and all. Okay? Thanks to all. Okay? And with these words, I can say that the webinar is over. Thank you one and all. Thank you.
Thank you.
Thank you all.